will now begin the LigaChemBio Global R&D Day 2025. I am Jung Dae-young, head of the IRBD team, and I'll be your host for today. Before we officially begin, we kindly all our participants on site to make sure that your mobile phones are set to silent or vibrate mode. Today's event consists of three main parts. Part 1, titled Current and Future Development Roadmap, will cover the current status of our clinical development programs and our future plans. Part 2, Next Drivers in ADC and Immuno-oncology, will introduce our new ADC pipelines and updates on next-generation immuno-oncology drug development of LigaChem. Finally, Part 3 will present our global business strategy and corporate outlook. Let me now introduce our executive leadership present today. As your name is called, please stand up and give a brief greeting. Mr. Yong-ju Kim, CEO of LigaChem Bio. COO and CFO LigaChem Bio, Park Se-jin. Mr. Yong-nak Cho, Senior Executive Vice President, Head of Development. Senior Executive Vice President and Head of Global Business Development and President of ACB, Choi Jae-uk. Dr. Jung-chul Uk, Head of Discovery Research. Mr. Seung-deok Chang, Executive Director and Head of Management Support Center. LigaChem's Executive Director of New Business at Orion and Internal Board Member of LigaChem, Mr. Hyung-seok Kim. Mr. Seung-ho Lee, CEO of Daily Partners and External Board Director of LigaChem Bio. Last but not least, let me introduce our key research leaders. Dr. Chang-sik Park, Head of the ADC Research Center. Dr. Suk-kyung Jang, Head of the Immuno-oncology Center. The leaflet you received includes QR codes for downloading presentation materials and submitting questions. Presentation materials will be available for download after the morning and afternoon sessions. If you have any questions during the presentations, please submit them via the QR code and we will address them during the Q&A sessions. All the morning session will feature English presentations only. We have translation receivers placed in front of you. Please utilize them. We will now begin the event with opening remarks by our CEO, Mr. Yong-ju Kim. Good morning, ladies and gentlemen. This is our second R&D Day event. Thank you very much for the great turnout, and also I appreciate your great interest in this event. We were supposed to begin earlier, but we had a lack of communications with you, so I apologize for that. Today, as was introduced by our MC and host, so most of our discussions will be about our R&D except for our business plans. So briefly, let me summarize what we are going to cover. As you see here, except for the business plans, everything else will belong to one of these three categories. One of the most important part is that we are going to have as many pipelines as possible. So we are making very aggressive approach. There are 5 pipelines in clinical trials, a total of 27 will be under development, and about 15 will be IND stages. So we are going to actively pursue clinical trials. So we are making a multifaceted approach, and also we are going to develop business development. Not only ADCs, but also modulate payload-based ADCs. So we've been preparing for these for years already. At the World EC Asia Summit, there were 3 keywords. One is the ADC, a bispecific ADC. Second, and bio ADC, there was a third one, PDC, peptide conjugate. So peptide conjugate is emerging again. Many people have negative thoughts about peptide, but peptide has actually a lot of advantages. So I'm a bit hesitant to talk about this. We are actually preparing for this. For the next 3 years, we are going to focus on these 3 categories. Aside from that, we are also focusing on novel targets, in addition to the clinically proven targets. So target development speed will be a lot slower down in the future. So I believe first-in-class novel targets will be identified soon. Second, regarding platforms, we continuously improve our platforms. Why? First, although we say customized linker, depending on the payload, properties and characteristics are different. So depending on the payload, we need to customize linkers. So when we have a new target, we are going to have a payload TOF. So although there's a mainstream movement, or statin, PBD, DNA-related stuff. So there are many different types. And also, we need completely new payloads. So if you continue to use one particular payload, it will develop resistance. So you need to change a payload. Since we are dealing with many different payloads, we need to develop the linkers, relevant linkers. So we have to continuously improve them. Second, I call it NOTA. This is very important. So far, there have been high DAR. So as you attach more payloads to antibody, the original properties of antibody will be lost. So PK will be worsened. So if you double DAR, that doesn't mean that efficacy will increase twice. It might rather increase toxicity twice. So low DAR, if you do that, TI will be further widened. So we are going to utilize a low DAR. Of course, we can attach many drugs to antibody, but we are trying to pursue low DAR. And committed therapy, that's very important. ADC and IOC, there have been many clinical trials. So there should be a scientific rationale. So because of that, we are developing many different ADCs and pipelines. So we are studying many different studies for new mode of actions, for combination therapies, new payloads. Since the founding of our company, this is our ongoing task. There are only 3 payloads clinically, so we need to identify a new payload, although it's arduous and lengthy process, but it's a must. Because if you continue to use the same type of payload, it will develop resistance. So not only STING. So We have all the backgrounds related to IO, and we are developing small molecules related to IO. So together with our partners, we are working together. Sometimes we already developed enough new mode of vaccine, new payload. We are identifying them and developing them. We will continue to do so. Regarding TPD, there's a limitations of TPD, especially for hematologic cancer. It can be utilized a lot. So for many different angles, we need to develop TPP, and we are working hard to develop TPPs from many different perspectives. So I believe our speakers will discuss that, especially in the later part of our event. So we are developing, we are growing, we are running our business. And regarding our business development plans, Mr. Che will give you details about it. Thank you very much.
Let us move on to the first presentation. As part of the current and future development roadmap, our first presenter is Robert Lutz under the topic of the ADC explosion, the expanding landscape of ADCs. Let me introduce Dr. Lutz prior to his presentation.
러츠 박사는 당사의 임상개발 부문 최고
Dr. Lutz is
자문위원이자
our Chief Clinical Development Advisor and currently serves as the CSO of our partner,
이전에
Iksuda
현재는
Therapeutics.
에보비사로 인수된
He is
이뮤노젠에서
a
23년간
leading
재직하며
authority in the
난소암
ADC
치료제인
field,
엘라히어,
having spent
유방암
23
치료제인
years
테사엘라 등
at
FDA
Immunology,
승인 ADC 치료제 개발을 주도하신
now
ADC
covered
분야의
by
최고
AD,
전문가이십니다.
where he led the
러츠
development of
박사는 오전 세 번째 발표를
FDA-approved
진행하실
ADC,
예정이어서
such as
Q&A는
Elara for
러츠
ovarian
박사의
cancer
세
and
번째
Casella for
발표가
breast
끝난 후
cancer.
한 번에
He's
진행하도록
also
하겠습니다.
going to give us third presentation,
Dr.
so
Lutz,
there will be Q&A session at the end of the part
I think
one.
that Mr. Kim already finished his time, so you got extra time. So,
Oh, great.
yeah, please start your presentation.
I will do. I'll look to share my slide right now.
Not yet.
It takes 30 minutes to
It's
go
okay
from
now.
Boston to Seoul, I think. Yes. Do you see my title slide?
You can see clearly.
Oh, great. Well, hi everyone. It's my great pleasure to speak to you all today, and I apologize for not being able to present to you in person. In this presentation, I would like to provide an overview of the expanding landscape. Just one second, technical problem here. There we go. I would like to provide an overview of the expanding landscape of the ADC field and introduce some of the opportunities and challenges that we face in thinking about the development of new ADC therapies. I characterize the past 5 to 6 years in the field as an ADC explosion, as noted in my title. In large part, this is due to the success of Enhertu, a HER2 targeting ADC for the treatment of breast cancer and other HER2-positive cancers. While interest in ADCs has existed for over 30 years, and I'll note I've been working on them almost that long, it was Enhertu that redefined what scientists considered could be possible for ADCs and has led to an explosion in effort and interest in the field. One way to look at the explosion is to map the number of clinical trials started per year. And starting as early as 2000, the number hovered between 10 to 15 clinical starts per year. A slight inflection came with the first ADC approval for Etcetras, and as clinical data emerged from clinical trials for TDM1 or Kadcyla, again, some increase of interest. But it was emerging clinical data from Enhertu that really put the effort on the modality into overdrive. Additional approvals have continued to feed that momentum. So the profile is similar if you look at the number of ADCs entering their first clinical trials, with over 62 ADCs starting clinical trials in 2024. This includes a surge of ADCs with topo-1 inhibitor payloads following the success of Enhertu, which is comprised of the topo inhibitor payload deruxtecan. In fact, most ADCs in the over 1,600 clinical trials currently ongoing use either a tubulin or topo-1 inhibitor as their payload. And this includes about 80 ADCs in phase 3 clinical trials. This large number of ADCs in late-stage trials predicts a continued substantial number of ADCs over the next several years. The excitement in the ADC space has not gone unnoticed by private and public funding interests, as well as interests in licensing and M&A opportunities. The number of ADC deals, either technology or asset licensing, or M&A deals, have averaged about 100 per year over the last 4 years, with total deal values reaching between 20 and 150 billion US dollars. And the global footprint of companies involved in the development of ADCs continues to grow, with over 1,200 biopharma companies currently involved in ADC R&D across all sizes, from small biotech to large pharma, with the top three locations for ADC R&D companies being the US, China, and Korea. So what's the big deal about the ADC space? Why is the commitment to the modality worth it? What is unique about ADCs that support continued investment in the space? So first of all, I think it's important to recognize that cancer still has an enormous unmet medical need. And even with all the advances in cancer therapies, most patients with cancer still die of their disease and spend the rest of their lives dealing with their disease and the side effects of their efforts to fight the disease. But still, why ADCs? I think the simple answer to that question is that ADCs are one of the few modalities that rely less on the biological properties of their targets and more on the characteristics of their design. This is drastically different from other modalities such as immune checkpoint modulators or other unconjugated antibody therapeutics. For this latter group, success is largely dependent on the biological hypothesis being correct. Is this target good for immune checkpoint inhibition or the like? If that hypothesis if that hypothesis is correct, and if I block the function of this protein, something good will happen. But if the hypothesis is not correct, there is very little learned to help with the next attempt. All that meant was my hypothesis about the biological nature of that target was not appropriate. And so it's back to the beginning. For ADCs, however, the designability of the modality means that prior experience increases the probability of success for next generation compounds. We can understand what the limitations of the current generation compounds are and innovate to find improvements that improve outcome. So, in other words, the strength of ADCs is that they are designable drugs. And indeed, while they're complex molecules, this complexity offers vast opportunities for optimization and differentiation. So new concepts, technologies, and designs are emerging all the time to address the shortcomings of prior design and exploit the learnings of the preceding generation of compounds. And in fact, the field is addressing all aspects of ADC design. This may include discovery of novel targets, for example, novel epitopes, or understanding the properties of ADC targets to manipulate them in a different way. It may include novel innovations for the binders, the antibodies, the alternative types of binders. Also, improvements in bioconjugation technologies for ease of development, improved biophysical properties, and improved stability. Novel linker chemistries are being developed to influence and improve the properties of the ADCs, for example, impacting its interactions or impacting the way in which it's activated, that we'll hear about more tonight. And it also may include improvements in payloads to overcome resistance, improve tolerability, and in particular, of interest to me, explore new mechanisms of action. And the importance of ADC design is no longer a hypothesis or an assertion. The impact of ADC design has been clearly validated in the clinic. And many of you who have been following the field will recognize this example. A simple comparison of TDM1 and TDXD highlights the importance of ADC design. TDM1 was first approved in 2013 with currently about $2 billion in annual sales. TDXD followed with first approval in 2019, 6 years later, and sales are expected to end up well over $10 billion a year. Both are directed against the same target, HER2, and utilize the same antibody, trastuzumab. So beyond that, the designs are very different. And it's clearly in those differences in designs that one sees the difference in these compounds. For example, TDM1 uses lysine conjugation chemistries. TDXD is conjugated to cysteines using classic maleimide chemistry. TDM1 has a non-cleavable linker, while TDXD has a peptidase cleavable linker. One, TDXD shows bystander activity, which may improve the activity in heterogeneous tumors, while TDM1 has little bystander activity, but that may provide improved tolerability compared to other designs. And potentially the most important difference for these two ADCs is that TDM1 has a tubulin inhibitor payload, with TDXD delivering a topoisomerase 1 inhibitor payload. The impact of the differences in the ADC design are profoundly clear when comparing the anti-tumor activity, with TDXD having a much higher response rate and progression-free survival in a head-to-head clinical study. They're not really very close, as it turned out. This level of response for TDXD at the time was unprecedented, particularly for solid tumors. But it is important to note that this improved clinical activity did come at the cost of significant hematologic, lung, and GI toxicity. And this latter fact highlights the need for continued innovation in the ADC field. So, what about other aspects of ADC development? And let's take a look first at the landscape of ADC targets that are being investigated. It's quite impressive, actually, the number of targets that have been evaluated over the years for ADCs. Over 100 targets are currently being explored clinically by at least one clinical ADC, with over 200 targets having been explored across across time by at least one ADC. Now, some of the ADCs to these targets have been discontinued, and many discontinued targets have no current disclosed efforts. But interestingly, many targets with successful clinically validated ADC programs have had discontinued programs in the past as well. This again highlights the importance of design, innovations, and learnings from prior generation molecules. So just because a target didn't work with an early ADC doesn't necessarily mean it's not a viable or valuable target with improvements in ADC design technologies. Importantly, also, most clinically validated targets have multiple active programs in development, where developers are moving forward with innovations designed to improve the outcome for patients, even where an ADC is already approved. So conceptually, we still believe that we can do additional design innovations and make ADCs that work even better. An example of this kind of innovation cycle is that HER2 and TROP2 remain the most studied targets even now, likely due to the high level of clinical validation and an understanding that continued improvements are needed and possible. That all comes back to the power of ADC design. Oops. Here we go. The payload landscape is also a dynamic one. There are over 3,400 disclosed ADCs in development. Leveraging the success of ADCs with tubulin and topo-1 inhibitor payloads with these MOAs still dominate the cytotoxic ADC class. Other DNA targeting payloads and radioimmunoconjugates are also growing in popularity for ADC design choices, and non-cytotoxic payloads are also advancing, with oligonucleotide payloads in particular being an active area. Though tubulin and topo-1 payloads dominate the development space, a variety of payloads have been used in ADCs that have gained approval, as well as a variety of different linker types, ranging from non-cleavable to linkers cleaved by different mechanisms. So while there's a great focus on tubulin inhibitors and topo-1 inhibitors, we have seen DNA alkylators, DNA cross-linkers be incorporated in designs that provided drugs that led to drugs that were approved. But once again, innovation is needed for the next generation ADCs. There are significant limitations with the current and prior generation ADCs that still limit clinical outcome. For example, regarding efficacy, not all payloads work in all indications. A good example of this is tubulin inhibitor payloads in cancers with spindle checkpoint mutations. Those mutations render those cancers very resistant to tubulin inhibitor payloads. Or the emergence of drug resistance can limit the activity of follow-on therapies. Cancers can become resistant to similar MOAs independent of the target use, so new payloads with alternate MOAs are needed. I'll touch on that again. Or potency versus tumor penetration. There was a big push to think about high drug to antibody ratio designs, but it's actually important to match the payload potency and the drug to antibody ratio to achieve antibody doses that are sufficient to drive tumor penetration. And often designs that have payloads that are too potent and too high a DAR don't end up delivering payload well to their tumors. Toxicity still appears to be the major limitation to increasing dose and efficacy. So suggesting that still safety, toxicity, is still a major design consideration when thinking about innovative chemistries and technologies. With regard to toxicity, traditional ADCs are limited by their treatment emergent adverse events. And these common toxicities are cytopenias, neuropathies, GI toxicity, lung toxicity, mucositis, rash, and others. And most ADCs still are dosed near or at their MTD, which suggests that improvements in tolerability would actually lead to improved outcomes. We're not reaching maximal anti-tumor effects. We're reaching maximal tolerated doses instead. So clinical data is now emerging from trials with new generation technologies, such as you'll hear about today from LCB, and continued innovation is still needed to improve these clinical outcomes. One key area for continued innovation is novel payloads. ADC sequencing will become the major development challenge in the future and was a key message at ASCO and ESMO over the past year. By sequencing, I'm referring to what therapies can be used following progression from an earlier line therapy. Much of the concern is that current data suggests that an ADC that follows another ADC with the same MOA shows relatively poor performance and independent of the target used, while good performances are seen when ADCs are sequenced with alternate MOAs. And over the next several years, as the landscape data shows, most indications will have at least one approved ADC with a topo-1 or tubulin payload as a standard of care. Therefore, new payloads with alternate MOAs will be desperately needed. Importantly, emerging novel ADC therapies, such as those that will be presented today, will help mitigate the risk of developing ADCs with new payloads. The improved properties provided by advanced bioconjugation, advanced linkers, and other design elements will contribute to the probability of success for novel payloads. So these technology innovations become even more important as they get applied to new and emerging other technologies. So all aspects of ADC design need to incorporate optimized technologies to continue the march toward better and better therapies. I want to thank you for listening.
Dr. Lutz, thank you. Thank you for your insight looking at this industry. Your Q&A session will be covered after your second talk.
I look forward to it. Thank you.
이어서 스테픈 슬로컨 박사의 LigaChem
Next, we
Biosciences
will hear
Platform
from Dr.
Technology
Stephen Slocum,
enables
presenting
differentiated
LigaChem Biosciences Platform
strategy
Technology
발표를
enables
진행하겠습니다.
differentiated clinical
Hello, Steve.
strategies.
Good morning.
Thank you for your presentation today. I will now introduce you to the audience in Korean. 슬로컨 박사는 당사의 보스턴 자회사인
Dr. Slocum works at ACB, our Boston-based subsidiary, and serves as the project lead for LigaChem Bio's ADC clinical pipelines, including the TROP2 ADC licensed to Janssen and the co-developed B7H4 ADC with NextCure in the US, which is currently being developed.
20년
So he's in
이상의
charge
항암제 분야
of
개발 경험을 한 전문가입니다.
project development. He is a
그럼
seasoned
발표
expert with
시작하겠습니다.
over 20 years of experience in
Slocum,
oncology drug development.
Please begin your presentation.
안녕하세요 and
Good
감사합니다
morning, ladies and
for taking the
gentlemen.
time
Thank you very much.
and giving me the opportunity to share updates on the pipeline. So I'll take some time today to discuss LigaChem Biosciences Platform Technology, enabling differentiated clinical strategies. And in the conversation, we'll address LCB84, LNCB74, and LCB02A. Just an outline of the conversation. We'll start off with a general overview of LCB's technology, as I feel that discussing the core technology and the manner by which it increases preclinical tolerability and efficacy, thereby widening the therapeutic index, is very important to understand the clinical strategies that we've undertaken. So LCB's technology improves patient tolerability and does this through three different aspects. Today, we will talk about two of them. The first is our bioconjugation platform, Conjual, and the second is our linker technology. A brief overview of Conjual. This is our bioconjugation system. It's site-specific and homogeneous. So it is achieved by engineering in a small amino acid sequence on the C-terminus of the light chain. This is then specifically functionalized via an enzyme, adding a small inert chemical isoprenoid substrate to the C-terminus, again, of the light chain. This is specific to this particular amino acid sequence at the C-terminus of a protein. So we only see functionalization of this engineered site. This allows, again, for a very site-specific and homogeneous bioconjugation. The isoprenoid is then reacted with our linker payload, which is only chemically compatible with the isoprenoid, maintaining that site specificity and homogeneous approach to bioconjugation. LCB has a number of chemical moieties that will enable either a DAR of 2, 4, 6, or 8, depending on whether or not we add in a mono, di, tri, or tetravalent linker. The linkage itself is plasma stable, unlike previous generation thiol maleimids, which undergo retrochemical deconjugation in circulation and lead to a loss of payload. The linker also allows and enables efficient release of the toxin inside of the target cells with tumor-selective proprietary glucuronide trigger. The payload conjugation linker system is compatible to most payloads available, including LCB's proprietary PBD prodrug, which will come up later, microtubule inhibitors, other DNA damaging agents, topoisomerase inhibitors, and payloads that are more exotic. Moving on to our linker, the beta-glucuronide, LigaChem's proprietary beta-glucuronide linker, shorthand is LBG. The mechanism for cleavage and release is shown here. In short, beta-glucuronidase cleaves the sugar moiety within the linker, driving an electron transfer cascade and allowing for the liberation of the parental toxin. This linker itself is plasma stable and tumor labile. LBG is efficiently cleaved by beta-glucuronidase, which is shorthand also referred to as GusB. GusB itself is overexpressed in cancer cell lysosomes, driving efficient release of the parental toxin. The release of the parental toxin itself is important. This enables the native properties of the payload. So in the case of MMAE, this would enable both bystander effect and immunogenic cell death. Other mechanisms of conjugation leave residual atoms on the payload and change those properties. Beta-glucuronidase is active at an acidic pH. pH 6.5 is the optimal pH. This really mitigates any payload cleavage in circulation. So GusB is not commonly found in circulation, but if it were, it would not be active as homeostatic pH is 7.4. Beta-glucuronidase itself has a very interesting expression pattern. There's minimal expression in healthy cells compared to tumor cells, which reduces payload released if there is on-target uptake in off-tumor cells of the ADC. So this linkage both mitigates off and on-target toxicity by minimizing systemic loss and maximizing delivery to tumor cells. Beta-glucuronidase itself is expressed and increased in both the mRNA and protein level across tumor types. This covers both liquid and solid tumors, and we haven't found a tumor type that doesn't express beta-glucuronidase at this point. We believe this expression pattern is due to the inherent metabolic requirements of tumor growth, driving the need for metabolism of any available energy source. And as mentioned, this enables the selective release of the active payload at the tumor itself. As mentioned, this linker is serum stable or stable in circulation. One of the benefits of LCB's technology is that it's often evaluated by external partners. The experiments shown here on the top are data from a large pharma partner who evaluated the technology and bioconjugated the same IgG1, antibody X in this case, with either LCB's bioconjugation system to a DAR4 MMAE or the previous generation val-cit stochastic DAR4 MMAE. You can see across cyno, mouse, and human serum that the LCB ADC, when incubated in vitro, shows no change in DAR over 7 days, whereas the old generation val-cit bioconjugation system shows significant loss of payload even after 24 hours. This result is recapitulated in the cynomolgus monkey, as shown at the bottom, 2 different ADCs showing basically overlapping curves of ADCs and antibodies over time in circulation, demonstrating the lack of loss of payload systemically. The other side of this coin is that LBG releases more payload compared to traditional linkers within tumor cells. So again, these are 2 different large pharma partners who evaluated our technology. On the left, the pharma partner conjugated, again, antibody X with either a val-cit DAR4 stochastic MMAE or a DAR2 MMAE via LBG's bioconjugation system, or a DAR4 with LCB technology. You can see in the red line that an LCB system conjugated ADC with a DAR of 2 releases almost exactly the same amount of payload as a stochastic DAR4 val-cit conjugation. And when you move to the orange line of a DAR4 LCB bioconjugated ADC, it releases considerably more payload within the tumor compared to a val-cit ADC. This is also seen by a separate partner who basically ran the same experiment with a different antibody, looking at the amount of payload released with an LCB linkage in red versus a val-cit linkage in gray. Importantly, the bottom line there is an isotype non-targeting control. So this shows that the LBG system releases more payload on a per mole basis of ADC delivered into the tumor. So this technology here, the LCB linker technology, enables, again, a highly homogeneous ADC with consistent properties. So this abrogates concerns on variable DAR species driving different toxicities. There's no concern of high DAR species causing, severe tox, or low DAR species, not having efficacy. This removes concerns on variable PK between different DARs. Everything is the same that's administered. You can optimize your molecule to drive responses in the tumor rather than drive toxicity systemically. LCB utilizes a stable bioconjugation system. This minimizes systemic exposure of free payload and minimizes potential off-target toxicity due to systemic exposure of payload, and maximizes the payload that's delivered to the tumor. When you think about this, this is very important. We go to all the trouble of making ADCs. You want to have the ADC do its job and take the payload into the tumor and not lose it systemically and then lose efficacy because the antibody is no longer able to drive internalization of the payload into the tumor and cell kill. LBG itself is a tumor-selective cleavable trigger, which increases the delivery of potent parental payloads into tumor cells and mitigates on-target toxicities by a reduction in release of potential payload in healthy normal cells compared to tumor cells. Taken together, this greatly increases the therapeutic index by both increasing safety and potency of LCB ADCs. Moving on into LCB84, our TROP2 targeting DAR4 MMAE ADC, where we have TROP2 targeting ADC with an auristatin payload enabling a differentiated clinical strategy, and LCB's core technology really enabling the use of a potent auristatin payload against TROP2 where others have failed. TROP2 as an antigen is very attractive, but also very challenging. It's highly expressed in both tumor tissue and in normal tissue, which drives the opportunity to regress tumors, but also drives a lot of on-target toxicity specifically in the skin, esophagus, and oral mucosa. Speaking of the opportunities within the TROP2 space, this is an estimation of market caps across indications where TROP2 has a preclinical rationale or a validated commercial rationale and clinical rationale for efficacy. ADCs have been approved targeting TROP2 in non-small cell lung cancer, breast cancer, and bladder cancer. Drilling down into bladder cancer, TROP2 is expressed across every subtype of breast cancer, HR positive, HER2 negative, HER2 positive, and triple negative breast cancer. Looking at the treatment lines for those three subtypes, HER2 positive breast cancer, you can see second line has trastuzumab deruxtecan, which is in HER2, as Bob has recently mentioned, has a topo-1 payload. And third line has trastuzumab emtansine or Kadcyla, which is a microtubule inhibitor. HR positive, HER2 negative breast cancer also has Enhertu approved in second line, as well as Trodelvy or sacituzumab govitecan. Again, both of those are topo-1 inhibiting payloads. And in HR positive, HER2 negative also recently had Dato-A approved in the second line, which is a topo-1 inhibiting payload. And then triple negative breast cancer has sacituzumab govitecan approved in second line or Enhertu approved in second line. So again, a lot of ADCs in breast cancer, but all of them right now, almost all of them have topo-1 inhibiting payloads. So we think that there's a significant opportunity for a non-topo-1 ADC within the breast cancer space. As kind of alluded to earlier, there are a number of emerging challenges in treating breast cancer. And one of the major points of concern is ADC sequencing. When Enhertu was approved, the phase 3 trial demonstrated that you are able to target the same antigen with a different payload class and maintain efficacy. So here, all of the patients treated in DESTINY-Breast02 had already been experienced to Kadcyla, and Enhertu demonstrated excellent efficacy and was approved based on the trial. However, when you start looking at prior exposure to a topo-1 bearing ADC, followed by treatment with a subsequent topo-1 ADC, you see a different story. These are 3 different vignettes at ASCO or ESMO in recent years, demonstrating a drastic reduction in the efficacy of the second ADC bearing the same payload class in sequential treatment. So this, again, really highlights the need for an ADC with a non-topo-1 payload in the breast cancer space, with TROP2 as an attractive antigen to target. It is important to acknowledge that the TROP2 ADC space has a lot of assets in development, but the vast majority of those are topo-1 inhibitors at this point, with Trodelvy and Dato-A both having been recently approved. To the best of my knowledge, the only active tubulin inhibiting ADC under current clinical development is LCB84, really demonstrating the difficulty of driving forward an auristatin or microtubule payload within that space. To really highlight that, we can talk about a clinical vignette on a previous generation TROP2 microtubule inhibitor bearing ADC and its design components. This is RN927, an earlier ADC brought forward into the clinic by Pfizer, targeting TROP2 with a stable conjugation DAR2 oral 101, which is a potent microtubule or inhibiting auristatin derivative. This was linked with a valicit linker with a cathepsin cleavable release mechanism. When you compare this to LCB84, LCB84 has a stable conjugation that's also homogeneous to DAR4 MMAE with our LBG linker with beta-glucuronidase cleavable release mechanism. When you look preclinically, LCB84 demonstrates increased preclinical tolerability compared to RN927C. Cynomolgus monkey toxicity studies show that LCB84 is considerably better tolerated compared to the Pfizer asset, despite LCB84 having a DAR of 4 versus RN927C having a DAR of 2. It's important to note that both payloads on these ADCs demonstrate comparable in vitro potency. The HNSTD of LCB84 in the DRF study was 12 mg per kg, Q3 week times 2. In the GLP study, the HNSTD was 10 mg per, greater than 10 mg per kg. Q3 week ×2, with histopath findings showing only single cell necrosis at the infusion site. For RN927C, their preclinical research paper states that 6 mg per kg was tolerated in the cynomolgus monkey, with a number of histopath findings covering increased mitoses and single cell necrosis in multiple epithelial tissues in the skin, injection sites, oral mucosa, and esophagus and vagina. In their phase 1 starting dose in the clinic was 0.15 mg per kg every 3 weeks. And when you do the math according to ICH S9 guidelines, that means that their HNSTD in the GLP study was 3 mg per kg. So despite bearing twice the molar amount of payload, LCB84 is greater than 3-fold more tolerated in cynos compared to this asset. When you look at the performance of RN927C in phase 1, it began the clinical trial in August of 2014 and was announced as discontinued in June of 2016. This ADC escalated from 0.15 to 0.3, 0.6, 1.2, and 2.4 mg per kg with no concerns. However, when this ADC was escalated up to 4.8 mg per kg, 50% of patients saw DLTs, including considerable skin toxicity, grade 3 rash, grade 4 toxic epidermal necrolysis, grade 3 bullous dermatitis, and febrile neutropenia. Pfizer then de-escalated twice to try and reach an appropriately safe dose, but was unable to due to continuing incidences of DLTs, further rash, neutropenia, and mucositis. The clinical toxicity profile here mirrors the on-target TROP2 preclinical findings in the monkey, with incidence of skin and oral mucosal toxicities, both reported in the cyno and in the clinic. Importantly, LCB84 does not show significant skin toxicity in cynos at doses equivalent to 3-fold higher than what RN927C was able to be dosed. LCB84 demonstrates a significantly wider preclinical TI, and the phase 1/2 study is actively progressing within the clinic. LCB84 provides a significant clinical and commercial opportunity as TROP2 is a validated antigen target for ADCs. ADCs with a topo-1 payload class play a major role in the treatment landscape of all subtypes of metastatic breast cancer, of which TROP2 is an attractive target for treatment in 2/3 of. Datroway was also recently approved in mutant EGFR non-small cell lung, validating this antigen commercially as a non-small cell lung ADC target as well. Datroway, importantly, is a topo-1 inhibitor payload, so we may be able to enter that space fairly easily. Many additional TROP2 ADCs are being developed. However, the majority of these ADCs utilize topo-inhibiting payloads. ADC sequencing studies demonstrate that payload resistance is a major issue in breast cancer. However, with LCB84, we have the opportunity to sequence with a different payload class and maintain that high efficacy. There is a significant opportunity for non-topo-1 payload inhibitor TROP2 ADCs, with LCB84 demonstrating higher activity and superior tolerability compared to failed first-generation auristatin ADCs. Moving on to LNCB74, our B7H4 targeting DAR4 MMA ADC that's being developed in collaboration with NextCure. This ADC has an auristatin payload that differentiates itself in complicated indication landscapes and utilizes LCB's core technology to drive improved clinical outcomes against B7H4 expression tumors. This is a slide from the most recent ASCO discussing the unmet medical need for B7H4 expressing tumors. The antigen itself is expressed across a number of indications, including breast, ovarian, endometrial, ACC, non-small cell lung, biliary tract cancers. B7H4 has been shown to be a negative prognostic factor, and there's a significant unmet medical need across these tumor types. This is another opportunity to target breast cancer with a non-topo-1 payload. It's also an opportunity to enter into the ovarian space or endometrial space, or to some extent the non-small cell lung. B7H4 as an antigen demonstrates an acceptable level of expression in ovarian cancer. This is data looking across a number of different antigens. When you look at B7H4 in the upper left and compare it to FOLR1, which has successful ADCs developed against it, you can see that it mirrors the same level of expression. Importantly, on the right, B7H4 and folate receptor do not have overlapping expression within tumors. they have exclusivity. So we can target tumors that Elahere is unable to target and not have to worry about following a DM4 payload. There is competition within the B7H4 targeting space. Much of it is topo1 driven at this point, so we don't need to fully be concerned about it, though we are expecting to show efficacy and better safety profiles. What does currently exist, at least in ovarian on the right, is Elahere, which is approved. We don't feel that we have fully overlapping expression profiles, so there are opportunities for treatment there. And Mersana's XMT-1660 or amiltatug ledadotin, which is in phase 1/2 with an auristatin F payload. This is a different representation of that last slide, but we will talk briefly about Seagen's discontinued B7H4V ADC, as well as run a comparison against XMT-1660. This is a schematic of Seagen's B7H4 vedotin. This is a stochastically conjugated average DAR4 ADC with a peptide cleavable linker and an MMAE payload. This ADC has been recently discontinued in phase 1 following Pfizer's acquisition of Seagen. However, this ADC has shown that B7H4 is a viable target for an ADC as they have efficacy data across breast, ovarian, endometrial, and biliary tract cancer. This ADC has a published preclinical cyno-HNSTD of 5 mg/kg in comparison to LNCB74, which has a cyno-GLP HNSTD value of 10 mg/kg and demonstrates a superior preclinical efficacy compared to a biosimilar of Seagen's molecule. That data is shown here. You can see that LNCB74 demonstrates superior efficacy in preclinical models of ovarian and breast cancer when compared to Seagen administered in a single dose. LNCB74 demonstrates regressions and or complete regressions at half the dose of Seagen's molecule. Seagen B7H4V's biosimilar, when administered at 6 mg/kg, really wasn't able to fully regress either tumor, whereas LNCB74 had full regressions at 4.5 mg/kg in ovarian and 3 mg/kg in breast. XMT-1660 is shown here. It's a site-specific conjugated DAR6 ADC utilizing a protease cleavable linker armed with an auristatin analog payload. This payload is advertised to have limited bystander effect. Clinical dose escalation of XMT-1660 began at 0.2 mg/kg, indicating an HNSTD in the GLP study of approximately 4 mg/kg. LNCB74 again utilizes an MMAE payload, so that retains full bystander effect and should enable efficacy in more heterogeneous tumors and potentially drive deeper and longer lasting responses in the clinic. The cyno-GLP HNSTD for LNCB74 was again 10 mg/kg, which is 2.5 times more tolerable than the XMT-1660 molecule. This is the phase 1 dose escalation clinical activity data shared by Mersana at the most recent ASCO. You can see in the subtherapeutic dose range, there is no activity. In the intermediate dose range, there is some but little activity against a number of different tumor types. And in the high dose range of greater than 76.2 mg/m² per cycle, there is an acceptable level of activity. However, at that dose, there is an unacceptable level of toxicity. Notably here, proteinuria, which is a complicated toxicity for physicians to manage, which we think leaves the space considerably open for us to move in and demonstrate better activity and a better safety profile to leapfrog this compound. I am able to speak to some extent about the phase 1 dose escalation of LNCB74. We are rapidly progressing through dose escalation across a number of B7H4 positive indications, including ovarian, breast, endometrial, biliary tract, and non-small cell lung cancer. Our IND was accepted on the 10th of December of 2024, with first patient in 10th of January of 2025. The second dose level was announced as cleared mid-April, and the third dose level was announced as dosing in early May. We have the opportunity and expect to run backfill for both safety and biomarker cohorts to generate a robust clinical proof of concept data set which we expect to be available in the first half of 2026. In summary, B7H4 is overexpressed in many cancers, including ovarian, endometrial, breast, BTC, NSCLC, and ACC. Right now, within that space, Elahere is approved in platinum-resistant ovarian cancer, but there exists ample differentiation between high folate receptor alpha and high B7H4 expression. Most B7H4 and ADCs in development have topo-wide payloads, enabling mechanism differentiation by LNCB74. And within the target indications for B7 overexpression, breast, ovarian, non-small cell lung, and endometrial, there's considerable opportunity for microtubule inhibiting payloads. There are clear and expeditious clinical development paths in existence for LNCB74, as there is significant unmet medical need across these indications. And compared to other microtubule inhibitor-bearing B7H4 targeted ADCs, LNCB74 demonstrates increased activity and a wider therapeutic index. This wider TI is expected to translate to improved clinical outcomes for patients. And importantly, MMAE does not exhibit proteinuria as an adverse event in the clinic, thus de-risking the safety profile of this asset in comparison to XMT-1660. Moving on to LCB02A, our Claudin 18.2 targeting DAR4 topo-1 inhibiting ADC. Here, gastric cancer and other Claudin 18.2 target indications present significant unmet medical need, and LCB's core technology serves to mitigate on-target toxicity and drive responses. Gastric cancer provides a significant clinical opportunity. It is the fifth most prevalent malignancy worldwide. The burden of disease is unevenly distributed, with Asia having the highest incidence. And the global burden of the disease is expected to increase in the next 25 years. So a significant market with a significant number of patients to treat. Claudin 18.2 is expressed at high levels in gastric and GE junction cancers, with additional opportunities in esophageal and pancreatic cancers. Importantly, highlighting the unmet medical need here, advanced gastric cancer has a very poor prognosis with an overall survival of under 1 year. Claudin 18.2 itself is a stomach-specific isoform of the Claudin 18 protein. Its expression is restricted in normal tissue with high levels present in gastric tissue. However, in healthy gastric tissue, Claudin 18.2 is thought to be largely inaccessible within tight junctions between cells. Following transformation, the antigen is mislocalized to the basolateral membrane, becoming accessible to systemically delivered targeted therapies. Approximately 50% of gastric cancer patients have greater than or equal to 2+ IHC staining and greater than or equal to 50% of cells. So generally, if the gastric cancer presents as Claudin 18.2 positive, it is very positive and reasonably homogeneous in terms of target. Importantly, the majority of Claudin 18.2 positive tumors are HER2 negative. This avoids the concern with previous HER2 exposure and topo-1 payload-mediated resistance. Claudin 18.2 expression pattern and intensity is similar across geographies, so the gastric cancer presents the same opportunities across Asia, North America, and other major markets. The estimated market size for Claudin 18.2 indications is shown here, with gastric cancer, GEJ, and pancreatic as the major opportunities. These are the NCCN guidelines for treatment of gastric cancer, both first and second line therapies. Importantly, an anti-Claudin 18.2 antibody, zolbetuximab, in combination with chemo, has recently been approved as first-line therapy in Claudin 18.2 positive tumors, really validating the antigen as a target. But what I think is most important here is that if you are not a HER2 overexpressing gastric cancer or an obvious cancer candidate for immuno-oncology therapy, your opportunities are minimal and are mostly systemic chemotherapy, which really highlights the lack of options in treating this disease and the high unmet medical need. A note about zolbetuximab, the recently approved antibody. Again, this zolbetuximab validates Claudin 18.2 as a targeted therapy target. It's improved in combination with chemo for first-line therapy in Claudin 18.2 positive tumors. However, second-line therapies and beyond are generally lacking. And this, I think, really enables promise to a targeted cytotoxic approach. When you look at the approval data for zolbetuximab, it really provides a minimal PFS benefit. On the left is zolbetuximab plus FOLFOX. On the right is zolbetuximab plus CAPOX. Both of those regimes only give you less than 2 months of additional median progression-free survival. So this is demonstrating really the ability to easily move ahead of zolbetuximab in lines of therapy, given an appropriate level of efficacy. The Claudin 18.2 targeted therapy space is crowded. I won't address the monoclonal or bispecific antibodies here as we are making ADCs. There are some microtubule inhibitor-bearing ADCs in later stage clinical development. We don't expect to have concerns following those if needed. The antigen Claudin 18.2 itself is conserved following Claudin 18.2 targeting therapy. For the Topo1 space, SKB315 is in phase 1/2. However, Merck recently returned that asset back to Kelun, and I think that speaks volumes. IBI343 is in phase 2/3, but as far as published data on this asset, seemingly it is in one center in China at this point, so not a lot of clarity around that particular asset. I think also importantly, there is a significant concern of on-target stomach toxicity with this target, which bodes well for LBG's ability to abate toxicities on target. LCB02A is armed with a DAR4 Topo1 inhibiting payload via the LCB platform, bioconjugation, and linker technology. This is expected to provide considerable safety and efficacy advantage versus competitor molecules. LCB02A itself offers an excellent preclinical therapeutic index. There are preclinical PDX models, not CDX models, that show excellent efficacy with an MED of less than 1 mg per kg in tumors. And cynotolerability is greater than 40 mg per kg with no significant findings. IND-enabling studies are ongoing, with an IND scheduled for the first quarter of 2026, and phase 1 clinical studies are planned to be run across Asia and North America in clinical locations with a high incidence of GI cancers. This will enable rapid recruitment and progression through phase 1 in key markets for the disease, and generate proof of concept across geographies to build strong justification for advancing and partnering the asset. With that, thank you all for your time.
Dr. Slocum, thank you for your excellent talk. 지금부터 Q&A 시간을 갖도록 하겠습니다.
Now we are going
Q&A는
to start a
당사의
Q&A
최재욱
session.
수석부장과 함께 진행하도록 하겠습니다.
Mr. Chejeo
Dr. Slocum, we will now move
participate in this
move on to the
Q&A
Q&A session. Dr. Che will lead. 발표관에 들어온 첫 번째 질문 보도록 하겠습니다.
Let's look at the first question.
스티브가 답변을 할까요? 제가 할까요? 제가 해도 될 답변일 것 같습니다.
Should I answer
Steve,
or
I'm
Steve
going
will
to take this
answer?
question.
Sure.
질문은 한마디로 지금 요즘 표현으로 중국발 ADC 쓰나미가 몰려오고 있습니다.
So there's a lot of ADCs being developed by China. A lot of trials are happening in China. What are the advantages of LigaChem's platform? It can actually drastically reduce toxicity. China biotech made ADCs also show great clinical results, according to the question. So can we actually maintain global competitiveness? It's a very critical and practical question. Let me answer this way. Shortly, Dr. Bob Lutz will share our HER2 data. But before that, when you look at the China's clinical outcomes, as was mentioned by Bob, among all the ADCs, regardless of targets, it is if you have to choose, the number one ADC is NR2, but it has the toxicities. It has blood toxicity in 70 to 80 patients, but it's very critical. About 60 to 70% toxicity is acceptable. But when you look at our HER2 ADC's result, grade 2 or 3 is less than 10%. When you look at the overall toxicity of our drug is just half of other competitors. But you're saying China-made ADCs show really great toxicity outcomes. But when you look at all the clinical data, so grade 2, 3, or 4 is less than 10% in our drug, but I didn't see that in other competitor drugs, including China-made ones. So China is actively developing drugs, but there's no innovations in China's ADCs. But they are combining the payloads and conjugation, but they don't have new payloads, no conjugations for their ADCs. So I can dare to say that we are above China in terms of safety and also in terms of innovation. We are ahead of China biotech companies. So regarding platforms, we are technically more above. Thank you very much. Second question. The question was about LCB84's development strategies and concerns about efficacy. Let me answer this way. Regarding Trop-2 ADC's results are pretty sensitive. LigaChem is Korea's flagship ADC, and it is obtaining a lot of licenses through global clinical trials. LigaChem is working with global partners, so we are following the global standards. With partners and licensees, we have contracts and we follow on those contracts. LigaChem LEDC, Johnson & Johnson's, Trop-2 ADCs. Unless they announce them, we are not supposed to announce them. That's our promise and contract with them. So our official answer is that we cannot answer to your question. But as was addressed by Mr. Steve, preclinical data already proved our excellence. So you can actually assume that even in clinical settings, the excellence will be proven. So our clinical trials are progressing very well, and the clinical trials results will be announced by our licensee, Johnson & Johnson. They will just follow their timeline. So according to the contract, we are not supposed to and we are not allowed to share the results yet.
Jay, I think I understood maybe 2% of that. But I think some of the failings of the Gilead and Daiichi Sankyo TROP2 targeting molecules can be attributed to their linker. Gilead and Trodelvy use an acid hydrolyzable linker where the payload is readily removed from the antibody. So if you're not really able to make a stable bioconjugation and drive payload into the tumor, you'll be able to see regressions and easier lower bars of disease like triple negative breast cancer when you're coming in against standard of care, which is just chemotherapy. but when you're starting to go into other more crowded disease spaces, you'll have trouble driving responses. Datraway also uses a GGFG linker, which is a slower to hydrolyze linker, which would hinder the amount of payload that you can deliver into cells in the tumor. Whereas I think GGFG would have a slower cleavage rate and less payload delivered than val-cit. And we've shown that LBG itself will deliver more payload than a val-cit on a mole-per-mole basis. So we have a strong rationale for driving more payload into the tumor, giving us a deeper and longer lasting response.
네, 뭐 스티브 답변 아주 간략하게 요약드리면 사실은 유사한 항암 효과를 나타낼 수 있죠. 그래서 이런 링커의 차별점으로 지금 다른 임상 헐투나 로어론에서 봤듯이 더 경쟁력 있는 약효를 기대할 수 있을 것 같습니다. 다음 질문.
답변 감사드립니다. QR로 질문 못 하신 분 중에서
Thank you very much. If you didn't have a chance to submit your question via the QR code, you can actually raise your hand on site. No question, then let's move on to the next question. Let's look at the second question. So I'm reading the question because it's quite long. I think it overlaps with what Steve already discussed. In a nutshell, if you have toxin A ADC, it's better to use toxin B. Why LigaChem continues with toxin A, although there's already toxin A ADC?
Sure. I just lost the question. That's fine. I think there was a note in there about systemic chemotherapy utilizing topo-1 inhibitors and that being a concern for cross-resistance with a topo-1 inhibiting payload with ADCs. That has interestingly not been the case. There are a number of solid tumors that are treated with topo-1 inhibitors as systemic chemotherapy that do not show resistance to ADCs armed with topo-1 inhibitors. The same is also true for microtubules. There are a number of taxanes used as free payloads and other microtubule inhibiting payloads that don't develop resistance to ADC payloads.
뭐 간단하게 말씀드리면은 그 ADC의 경우에는 페일로드 A 다음에 또 페일로드 A를 사용하는 게 좀 가능성이 떨어지지만 사실은 스몰 모레큘을 A를 쓰고 그 다음에 그와 유사한 페일로드 A를 가진 ADC를 사용하는 경우에는 그렇게 사실은 약효가 떨어지지 않습니다. 뭐 예를 들면은 저희가 이렇게 많이 아는 실제로 병원에서 다들 아시겠지만 first line은 대부분 다 뭐 스탠다드 오브 케어가 키모테라피입니다. 키모테라피에는 당연히 마이크로튜브 이미디터가 들어있는 경우가 되게 많죠. 근데 거기서 약을 조금 투여받다가 잘 안 들으면은 아직도 ADC는 대부분 세컨 라인.
So we are basically looking at the most standard of care. So they are microtubule inhibitors. So when you look at the ORR, so they are not for naive patients. So they already treated patients come back and they are treated with ADCs. There's a payload A ADC, payload B ADC. So which one is better, A or B? This is a big question. In order to make story simple, So we only announce the candidate ADC. But actually we are producing multiple payloads, A, B, C, D's. Efficacy and toxicities are being studied for them. And based on that, the best therapeutic index ADCs are selected and they are injected as a candidate drugs in clinical trials. We don't just simply go that because there's A, we do B. It's not simple like that. So due to time constraints, Q&A session will be concluded here. So you have 2 QR codes, one for download, 2 for question. But right now, both QR codes can be used for submitting questions. So after the morning session ends, one of the QR codes will be turned into a download QR code. Now, let's move on to the presentation by Dr. Lutz.
Here is going to talk about the case studies from Iksuda Therapeutics of LCB technology in action.
For your second talk today, please begin your presentation.
Great. I will share my screen here.
Yeah, everything's okay now. You can start.
Great. Well, hello again, everyone. I'd like to take you through, hold on. Great. I'd like to take you through 2 case studies of ADCs being developed by Iksuda Therapeutics that help to demonstrate the power of LCB's ADC technology. The first program is we call IXO14, a HER2 targeting ADC for the treatment of HER2 expressing cancers. And the second program is IXO4, a CA242 targeting ADC for GI toxicities. Iksuda Therapeutics is an ADC development company headquartered in Newcastle, England, with its development activities directed out of our offices in Boston, Massachusetts. Our most advanced programs are ADCs that contain LCB technology. The emerging data from these programs and others illustrates the power of LCB's technology for improving preclinical and clinical outcomes for efficacy and safety. I'll provide an overview of the findings to date for the IXO14 and IXO4 programs, and mention some findings for our IXO3 program as well that's shown in the table here, a CD19 targeting ADC with LCB technology that led us to the development of IXO4. We'll start first with IXO14. As I mentioned, this is an anti-HER2 targeting ADC with an MMAF tubulin inhibitor payload for the treatment of HER2-positive cancers, including breast, lung, and GI tumor indications. So a first question to address is why another HER2 targeting ADC? While TDM1 or Kadcyla, a HER2 targeting ADC with a maytansinoid tubulin inhibitor payload, was the first ADC to be approved for the treatment of a solid tumor indication, it was Enhertu, or T-DXd, as I described earlier, with its high response rates and durations of response that really ignited global interest in the ADC modality. Enhertu continues to show impressive response data and is likely to become first-line therapy for patients with metastatic HER2-positive breast cancer, as well as other HER2-expressing cancers. However, there are 2 major challenges still to face. One is that while T-DXd is very effective, it has a challenging toxicity profile, with many patients having to cope with significant GI toxicities during therapy, such as nausea, vomiting, and diarrhea. These toxicities often lead to dose delays and discontinuations. Secondly, the majority of patients ultimately progress during or following T-DXd treatment and require additional therapies to control their disease. And emerging clinical data suggests that the best clinical outcomes are achieved when therapies with different mechanisms of action are used in sequence in treatment paradigms for patients. In other words, treatment outcomes have been disappointing when an ADC with a topoisomerase payload is used after another ADC with a topoisomerase payload. So the best outcomes have been seen when sequencing ADCs with alternate MOAs. These findings support the development of a best-in-class HER2 targeting ADC with a non-topoisomerase payload. The relatively low response rate of T-DM1 in second or third line metastatic breast cancer and its lack of sufficient activity in other HER2-positive tumor indications, such as gastric cancer, supported the concept of a next-generation ADC with a tubulin inhibitor payload that is more active than T-DM1, and preferably with a toxicity profile that is better than T-DXd. So IXO14 is a novel third-generation HER2 targeting ADC with a modified trastuzumab antibody engineered to be compatible with LCB's site-specific stable conjugate bioconjugation technology. And the ADC incorporates LCB's stable glucuronide cleavable linker with an MMAF auristatin tubulin inhibitor payload. Upon entry into a HER2-positive cell, the linker is cleaved by glucuronidase, releasing the potent payload, leading to cell death. Iksuda has licensed IXO14, now with the generic name of caxmotobart N2-dotin, but I'll keep referring to it as IXO14 for ease, for global development with the exclusion of China and Korea, and is currently conducting a phase 1 clinical trial. Fosun Pharma has licensed IXO14 for development in China under the name FS1502, and Fosun is currently in a phase 3 registration trial for advanced HER2-positive breast cancer in China. One important contribution of LCB's ADC technology in the design of IXO14 is the improvement in the toxicity profile of this MMAF ADC. Traditional MMAF ADCs most of them did not advance into late-stage trials due to problematic toxicities, the most notable one being corneal toxicity. One ADC that has successfully been developed is the BCMA targeting MMAF ADC from GSK called belantamab mafodotin, or Blenrep, for the treatment of multiple myeloma. But comparing the toxicity profile of IXO14 to this traditional MMAF ADC clearly demonstrates the impact of LCB technologies. Drawing from Fosun's phase 1 data presented at ASCO in 2023 on approximately 100 patients at their recommended phase 2 dose, a substantial improvement in the toxicity profile compared to Blenrep was demonstrated. While IXO14 shows excellent activity in its indication, it does so with markedly less thrombocytopenia, GI toxicity as exemplified by cases of nausea, and drastically reduced corneal toxicity and impact on vision. Same payload, much improved tox profile by improved ADC design chemistries. Looking then at IXO14 compared to TDM1 and TXD, we again see the impact of LCB's ADC design innovations on outcomes. While the target and antibody are the same, IXO14 has design changes in bioconjugation with site-specific homogeneous enzyme-based conjugation compared to non-site-specific through lysines or hinge cysteines of the other ADCs. IXO14 uses a low DAR approach to improve tumor penetration, and it has a linker that has improved selectivity between tumor and many normal tissues. It also has minimal bystander, thereby reducing toxicities due to free payload, potentially leading to reduced neuropathies typical of a tubulin inhibitor ADC. The result is a highly active ADC with activity similar to Enhertu in phase 1 patients, but with a notable improvement in its toxicity profile, with lower rates of lung findings, lower GI toxicity, and lower blood cytopenias. This encouraging clinical profile was the driver for Iksuda to initiate their own phase 1 to further explore the dose relationship of IXO14 for efficacy and tolerability. We opened the trial in Australia and have recently expanded to sites in Singapore, New Zealand, and the US. The phase 1 design includes a typical 3+3 dose escalation phase with the addition of enrichment cohorts at selected dose levels. This part 1 of the trial is aimed at identification of the MTD and/or a recommended phase 2 dose, providing sufficient data to allow selection of a dose for a dose expansion phase, or part 2, that will look at specific patient populations, currently anticipated to be HER2-positive breast cancer in patients previously treated with Enhertu, HER2-low breast cancer, HER2-positive gastric and GE junction cancers, and a basket cohort for HER2-positive indications beyond breast, gastric, and GE junction. A snapshot of the preliminary data from Iksuda's phase 1 shows 1, we're observing responses at multiple dose levels, including our first dose level of 40 mg per meter squared. And I'll note here that we're using body surface area dosing that helps normalize ADC exposures across patients with varied body mass indices. And for your guidance, 40 mg per meter squared is comparable to dosing at about 1.1 mg per kg in a patient with a typical BMI. Secondly, we've seen responses in multiple HER2-positive indications, including breast, esophageal, gastric, endometrial, GE junction, lung, ovarian, and gallbladder cancers, with the majority of patients that have shown responses and/or stable disease still on treatment. Thirdly, as use of Enhertu has only recently become common in Australia, we have had only a limited number of post-Enhertu patients thus far. However, recent data has been encouraging, with 4 of 5 post-Enhertu patients showing a response in the higher dose cohorts, and the 5th patient still showing stable disease. The opening of centers in Singapore and the US is intended to increase the number of post-Enhertu patients available for the trial, and we will be presenting a full clinical update at ESMO 2025 in Berlin in October. The safety profile observed so far in the Iksuda phase 1 is very consistent with the observations disclosed by Fosun from their clinical experience. The main treatment-related adverse events are generally low-grade, dry eye, dry mouth, and hypokalemia, all of which can be mitigated somewhat by simple interventions such as eye drops, mouth rinses, and potassium supplements. Unlike traditional MMAF ADCs, there has been no high-grade thrombocytopenia or corneal toxicity concerns, and minimal GI or lung toxicity compared to Enhertu. Again, a full update on the safety profile will be presented at ESMO. Overall, Iksuda has seen strong phase 1 clinical data with responses seen in post-Enhertu patients and more activity than expected for TDM1 in this population, supporting the potential for IXO14 being an appropriate second or third line treatment option in HER2-positive breast cancer. Additional clinical evaluation will be needed to understand the potential of IXO14 in other HER2-positive indications, but early data is encouraging. And as already noted, IXO14 is well tolerated with a manageable tox profile that is significantly different than ADCs with topo-1 inhibitor payloads. I'd like to switch the focus now to another Iksuda asset that utilizes LCB ADC technologies. Our CA24 targeting ADC program grew out of our early experience with another LCB ADC asset, IXO3. IXO3 excuse me, is a CD19 targeting ADC which incorporates LCB's prodrug DNA crosslinker technology. The prodrug approach incorporates LCB's glucuronide trigger chemistry for both the release and full activation of the payload. As exemplified by IXO3, this technology greatly improves the therapeutic index of this highly potent payload compared to prior efforts. The comparator for IXO3 is an ADC developed by Seattle Genetics. SGN CD19B was a CD19 targeting ADC conjugated to a traditional PBD DNA crosslinker called tolurine. CD19B used a traditional Val-Cit cathepsin cleavable linker, and development was ultimately discontinued due to poor tolerability. IXO3 with LCB's ADC technology shows comparable preclinical efficacy to CD19B. However, in monkeys, its HNSTD is 5-fold higher. A comparison of the ADC's ability to kill cancer cells in mouse xenografts versus depletion of CD19 positive normal B cells in the monkey provides a window into the power of the glucuronide prodrug format in IXO3. While CD19B depletes normal B cells at a dose even lower than the dose needed to kill cancer cells, IXO3 causes only partial B cell depletion at a dose over 10 times greater than CD19B. This difference is believed to be due to the differential expression of glucuronidase in malignant versus normal B cells, and may be reflective of low glucuronidase expression in many normal tissues. So, with low beta glucuronidase, it's thought that the linker cannot be processed efficiently to cause cell death. IXO3 is currently in a global phase 1 clinical trial for patients with B-cell malignancies. But the data for IXO3 encourage us to ask whether LCB's technology would support use of this payload in solid tumor indications as well. Traditional ADC payloads such as tubulin and topo-1 inhibitor payloads have shown limited activity in hard-to-treat GI cancers, and a DNA cross-linking payload would be expected to be very active in these indications. However, high-potency payloads like PBDs have been limited by poor tumor delivery and poor tolerability. So the improved tolerability of LCB's prodrug approach may make solid tumor targeting a possibility. Given the high potency of the PBD platform, we sought to evaluate the possibility with a highly tumor-selective target, CA242. CA242 is a cancer-associated glycotope with high expression in a broad array of hard-to-treat tumor indications, but has very limited normal tissue expression, suggesting a low risk of on-target toxicity. So IXO4 is comprised of Iksuda's proprietary humanized antibody targeting CA242 with LCB site-specific conjugation, glucuronide trigger and prodrug chemistry, and a highly potent PBD DNA cross-linker. The ADC shows highly potent, target-dependent in vitro tumor cell killing, with the in vitro potency correlating well with target expression levels, with very low EC50s observed with cells expressing moderate to high levels of CA242. The ADC also exhibits strong bystander activity, where antigen-negative cells can be depleted in the presence of a low percentage of positive cells. No activity is observed with the negative cells in the absence of target positive cells, and this bystander activity can potentially improve the activity of the ADC in heterogeneous CA242 expressing tumors. IXO4 also shows strong in vitro activity across a range of models. However, initial studies were a bit surprising in that higher expression did not result in increased in vivo efficacy. In fact, the highest expressing models had the higher minimally effective doses. This inverse correlation is indicative of an antigen barrier effect. This effect is due to high target expression limiting the penetration of the ADC into the tumor. Rather, the ADC is captured on tumor cells that are closest to the blood vessels, as shown by the middle figure, the illustration on the left. This antigen barrier challenge is particularly important for ADCs with high potency payloads, as the high potency can limit the clinical dose of the ADC. This necessitates another approach to drive penetration. Investigation by Greg Thurber's laboratory at the University of Michigan and others have demonstrated that ultra-low DAR regimens by co-administration of the ADC with unconjugated antibody can help overcome the antigen barrier effect and improve ADC distribution within the tumor, and thereby increasing efficacy. This is particularly useful for antigens with high expression. And the immunofluorescence shown on the right clearly demonstrates the improved penetration and overall delivery with increased antibody dose. Evaluation of the efficacy of this ultra-low DAR approach with IXO14 shows how impactful it can be. In the study shown on this slide, with a high-expressing gastric cancer model, the original study showed that the MED, the minimally effective dose of IXO4 in this model, was 1 mg per kg single dose. Co-administration with 10 to 50-fold excess of unconjugated antibody improved the MED down to 0.4 mg per kg. This approach may not provide improvement for all targets or for all levels of target expression, though preclinical data has shown improvements in efficacy for a wide range of targets and may be an appropriate approach for many applications. For CA242, since the majority of patients that are CA242 positive have moderate to high expression, as exemplified by our models, we've chosen to take this ultra-low DAR regimen into the clinic. You see the percentage of patients that have moderate to high expression in the graph on the left. All of the blue patients would be those considered for this type of approach. About 80% of colorectal cancer, GE junction, gallbladder, cholangiocarcinoma, and pancreatic cancers show moderate to high expression of CA242. In gastric cancer, it's about 60% of patients that have high to moderate expression. This leads to a very large potential patient population that have failed frontline chemotherapies with this level of CA242 expression. Estimates of greater than 2 million patients may be expected to need second line or third line therapies annually that would have high to moderate CA242 expression. Interestingly, this ultra-low DAR approach is not dissimilar to what's being evaluated for radioimmunoconjugates, and the FDA has provided positive feedback to Iksuda regarding using this ultra-low DAR regimen for IXO4 clinical evaluation. IND studies are well underway, with a US FDA IND filing expected later this year. Our trial design will consist of a dose escalation phase, including enrichment cohorts at select dose levels, followed by a dose expansion phase to look more closely at selected indications, along with a basket cohort, very similar to what is planned for IXO14. And we're incredibly excited to bring this program into the clinic. Finally, I'd like to thank all the patients, their families, caregivers, and investigators for their participation in our clinical trials, and also the Iksuda and LCB teams for their efforts to support these programs. I thank you for listening, and we'll be happy to take any questions.
So 14 clinical data.
The question is not yet on my screen.
Okay, so let me read this for you, and then.
Now I see it.
Okay, probably this is the question you can answer.
Do you think? Yes. Even the experience so far with TDM1 suggests that prior therapy with Enhertu does not decrease the response rate to ADCs with a different payload, in the case of TDM1, a tubulin inhibitor payload. So our initial target product profile is to position IXO14 post Enhertu, and essentially go head to head with TDM1 or other HER2 targeting small molecule agents. I think there's a high potential, given what we've seen and the response rates, at least in phase 1, that are similar to Enhertu, and we'll know some about Fosun's phase 3 data over time, that IXO14 may also be competitive with current first-line standard of care, namely trastuzumab and taxanes. So I think we have an expectation that IXO14 could be positioned second line post Enhertu. Now, direct competition with Enhertu would be challenging, although it may be possible that a large strategic partner would be interested in taking on that role. But the high response rates for Enhertu do provide somewhat of a challenge to think about direct competition. But again, I think we're well positioned for second or third line therapy. I would say currently no other ADC has as favorable a response and toxicity profile with a payload other than topo-1.
Okay, thank you, Bob. Let me just translate your answer to Korean just for the next couple of minutes. 질문은 사실 오늘 발표 중에 뭐 제일 중요한 것 데이터인 것 같은데 NR2를 맞은 질문은 그러면은 이 리가캠의 익수다의 그 헐투 ADC를 과연 이처럼 NR2 트리트먼트 된 다음에 포스트 NR2로 할 것이냐 아니면은 헤드 투 헤드로 컴피티션을 할 것이냐 뭐 심플한 질문인데 사실은 굉장히 여러 가지 팩터들이 포함된 사실 답변을 드릴 수밖에 없는데요. 이제 밥의 앤서는 아까 밥이 설명했듯이 원래 마이크로튜블 이니비터를 포함하는 TDM1을 맞고 잘 안 듣는 환자들이 그 토포아이 페이로드를 가진 NR2에 의해서 치료 효과가 잘 나타났듯이 아마 비슷할 것이다. 그리고 실제로 이제 4명의 PR이 나왔으니까 그 입증은 됐죠. 그래서 NR2 다음에 우리 헐2가 들어가서 그건 충분히 경쟁력이 있다. 그래서 현실적으로는 지금 이제 임상에 들어가거나 혹은 허가를 받아도 이미 사실은 NR2를 맞은 환자들이 엄청나게 많을 거예요. 그러니까 첫 번째로 나이브한 환자들이 저희한테 오지는 않을 거니까. 그러니까 세컨 라인으로 포스트 NR2는 너무나 당연하게 차지할 것이다. 근데 이게 정말로 모든 회사들이 지금 찾고 있는 솔루션이거든요. NR2가 점점 더 매출도 많이 오르고 이제 인기 있어지면서 과연 그러면은 NR2 안 듣는 환자들은 어떻게 할 거냐? 그냥 터미널로 끝날 것이냐? 솔루션이 없는 상태에서 저희처럼 사실은 이런 임상 대회를 통해서 포스트 NR2가 솔루션이 있으면 굉장히 좋죠. 그 다음에 헤드 투 헤드 컴패리슨은 초기 단계에서는 사실은 챌린징 할 수도 있지만 익수다가 그 퓨처 뭐 파트너십 얘기를 잠깐 했는데 뭐 끝까지 사실은 나중에 막 대규모 삼상 같은 거를 뭐 할 수도 있겠지만 하시기는 현실적으로 힘들 테니까 만약에 미래 빅파마 파트너가 테이크오버를 해서 그런 거에 인트레스트 한다면은 실제로는 컴패리슨 스터디도 뭐 가능하겠죠. 왜냐하면은 아까 보여드린 대로 NR2는 여전히 굉장히 많은 부작용 때문에 환자들이 사실은 그 서퍼링하는 부분들이 있는데 여태까지 정말로 보여주지 않았던 아까 보셨다시피 뭐 구토나 뭐 모든 면에서 사실은 혈구 독성이나 이런 것에서 현격하게 낮기 때문에 그런 측면을 생각하면은 나중에 미래 뭐 빅파마라고 얘기하진 않았지만 그런 파트너가 헤드 투 헤드의 임상 디자인도 가능은 할 것이다. 이렇게 대답했습니다. 네.
예, 오늘은 데이터가 좀 짧게 나왔지만 자료에도 있듯이 에스모에서 한 번 더 발표할 예정입니다.
So there will be another presentation at ESMO. So detailed data will be presented at ESMO. We'd like to move on to the second question.
Bob, the second question is coming up.
Yep. I'm reading through it. Very
Okay.
interesting. Okay. Very, very interesting question. So I do believe that the payload plays the biggest role in and the ability to utilize that payload, as in the case of IXO14, since a traditional MMAF conjugate would probably not have been sufficient. It's payload MOA that I think is the biggest driver of next favorable outcome, more than the antibody or the data thus far, in large part driven by studies post in HER2, have suggested that following with the same payload, regardless of target, shows poor performance. deciding whether to follow with HER2 or a TROP2 ADC with a different payload, such as IXO14 versus LCB84, for example, is a question that's difficult to answer at the moment. Are not big
Hey,
enough
Bob, but
yet
it doesn't need to be 84. It can be like a Trodelvy or like a Daiichi Sankyo's
Well,
TROP2.
yeah. So any topo one payload after a topo one payload, the current data suggests that will show poor performance, regardless of target. So payload's the most important factor thus far in our understanding of how best to sequence ADCs.
Great. 이 질문도 사실은 굉장히 중요한 질문인데요. NR2에 대한 내성이 한마디로 항체가 중요하냐, 페일로드가 중요하냐 그 질문입니다. 그러니까 토포 I에 대한 내성이 암세포에서 생길 수도 있고요. 혹은 헐투 엔티젠 자체가 뭐 돌연변이거나 없어져 버리면 사실은 그 다음에 헐투 ADC를 처리해도 또 계속해서 내성이 지속될 거니까. 그러니까 근데 지금까지 임상 데이터들을 종합해 보면 지금까지 밥의 질문도 그렇고 전체적인 컨센서스는 페이로드가 더 중요하다입니다. 그 내성에 있어서. 그러니까 뭐 예를 들면은 100명의 환자가 HER2 ADC의 내성을 보이는데 그 중에 페이로드 내성이 더 많은지 아니면 HER2 자체가 뭐 익스프레션이 변하거나 어떤 게 더 많냐 보면은 실제로 페이로드 내성이 더 많다는 게 지금까지 임상의 데이터고요. 그래서 여기 보면은 만약에 HER2가 변한 거면은 트롭2 같은 거 트롭2 ADC로 그냥 처리하면은 그게 극복되지 않겠냐 사실은 질문에 그게 포함이 돼 있는데 밥의 앤서도 그렇고 지금까지 임상 데이터로 예를 들어서 내성 환자를 일일이 다 검사를 해서 이게 헐투가 바뀐 건지 아니면은 토포아이의 내성이 있는지 그거를 일일이 다 뭐 유전체 분석을 하거나 이래서 그 다음 단계의 약물의 임상에 사실은 들어가는 경우는 매우 드뭅니다. 그냥 안 들으면 NR2가 안 들으면 사실은 그 다음 약물로 진행되는 경우가 많거든요. 그래서 그런 경우에 지금까지 임상 데이터를 보면은 트로델비나 그러니까 NR2가 안 드는 환자를 트로델비나 트롭2 ADC로 처리를 해보면 반응성이 되게 안 좋았어요. 그 얘기는 토포아이가 들어간 ADC를 한번 한 환자들은 그 다음에 타겟은 다르더라도 똑같은 페이로드인 토포아이가 있는 경우는 사실은 잘 안 들은 경우가 많습니다. 그래서 이거에 대한 답은 페이로드를 사실은 바꾼 ADC로 트리트하는 것이 사실은 가장 좋다가 지금까지의 통계학적인 답변입니다.
예, 세 번째 질문 올려주십시오.
So we'd like to move on to the third question.
흰 글씨가 좀 잘 안 보이는 것 같아서 검은 글씨로
So
바꿨습니다.
we have changed the colors to make it more remarkable. Can you please take a look at the third question on screen?
Hey Bob, do you see the
Another great question. So one of the things that's emerging about HER2 is that there are very few patients that are actually HER2 negative. Although it is clear that low and ultra low activity of HER2 is probably driven by a lot of tumor microenvironment release of the topoisomerase payload. That being said, we are with IXO14, Iksuda and Fosun have seen activity in HER2 low. I wouldn't say we have experience with ultra low. So it's not clear that the post in HER2 market for compound like IXO14 would only be current HER2 positive high breast cancer. On the other hand, it is going to be important for ADC development in the future for ADCs to carve out more and more specific patient characteristics that will help differentiate from other next generation ADCs that are coming along with targets in certain populations and so forth. So, for example, post and HER2 market for HER2 breast cancer, HER2 high breast cancer, and potentially HER2 high gastric and esophageal cancer will be a higher than billion-dollar market in itself. So I think not a bad market for a second or third line therapy in that case. The plus side of MMAF not having bystander effect is it's less cell permeable. So in a target directed cell, in a target positive cell, it can accumulate to eventually the concentrations that are needed to kill the cell. So that in part compensates for lack of bystander activity. Is that you're not connected, you're not dependent on the kinetics of delivery, that you can take more time to deliver payload through with lower antigen density. The upside, of course, is incredibly important for tubulin inhibitors. One of the biggest clinical relevant toxicities of MMAE conjugates, which you don't talk about often because it's not part of the DLT window, is neuropathy. And when patients are treated with a vedotin ADC for multiple cycles, neuropathy is often an important clinical toxicity that has to be dealt with. MMAF is expected to show very limited neuropathy because the free drug is not permeable to nerve cells. So it is a balance.
Okay, great. 질문부터 뭐 잠깐 설명드리면 뭐 다들 이해하셨겠지만 MMAF는 전통적으로 굉장히 소수성이 높다고 인식되고 있어서 암세포에서 암세포를 죽인 후에 옆에 세포를 잘 못 들어가요. 그래서 이제 바이스탠더 이펙트가 적다고 하는데 이렇게 적으면 한마디로 HER2 익스프레션이 굉장히 나는 캔서에서는 옆에 암세포를 죽이는 거에 좀 제한적이니까 이렇게 HER2 low나 ultra low breast 캔서에서는 한마디로 LCB의 ADC는 잘 안 듣지 않겠냐 하는 게 뭐 이 질문의 일단은 요지고, 그렇게 되면은 포스트 NR2 마켓에서 HER2가 높은 환자에 사용된 포스트 NR2 환자에만 되지 않겠냐, 그래서 시장이 좀 줄어들지 않겠냐인데 밥이 조금 길게 설명을 했는데, 일단은 그동안 호주에서 진행된 임상에서 실제로 HER2 로도 포함이 돼 있었어요. 근데 HER2 로에서도 저희가 효과가 잘 나오고 있기 때문에 실제로는 아마도 포스트 NR2에서도 HER2 로에서도 저희가 뭐 기대할 수 있을 겁니다. 그래서 뒷부분에 설명한 거를 뭐 밥이 설명한 걸 앞부분으로 당겨오면 바이스탠더 이펙트가 없다는 게 굉장히 단점 같지만 일단은 독성의 장점이 있고요. 왜냐하면 옆으로 퍼지다 보면 결국은 노멀 셀도 죽이게 되니까. 두 번째로 바이스탠더 이펙트가 없다는 것의 장점은 뭔가 하면은 왔다 갔다 셀 밖으로 안 하기 때문에 MMAF는 축적이 많이 됩니다. 그래서 한마디로 블러드에서 ADC가 서큘레이션이 오래되면은 들어온 게 안 나가요. 예를 들어서 MMA 같은 거는 왔다 갔다 하고 나가게 되거든요. 그래서 뭐 같은 오리스테틴이라도 장단점이 있다고 이제 밥이 설명한 이유는 MMA ADC는 쓰면 항상 뉴로패티라는 게 나와요. 그러니까 신경계로 굉장히 아픔을 호소하는, 신경 말단 고통을 호소하는데 그 이유가 신경세포로 MMAE가 쑥쑥 잘 들어가서입니다. 그러니까 떨어져 나오는 프리 페이로드가. 근데 MMAF는 그런 게 없어요. 왜냐하면 바이스탠더 이펙트가 없으니까. 떨어져 나온 게 못 들어가죠. 대신에 ADC로는 한 번 들어가면은 안 나오고 그 다음에 ADC가 또 들어오고 하니까 바이스탠더 이펙트가 없다는 게 그냥 무조건 효능이 더 좋지 않음을 의미하지는 않습니다. 지금 밥이 설명한 대로 이런 이제 어큐뮬레이션이 되기 때문에. 그래서 결론적으로 말씀드리면은 헐투 로도 저희가 충분히 가능성이 있다고 생각하고 아마 포스트 NR2 마켓에서 무조건 헐투 하이로 국한되지는 않을 것이다. 근데 플러스 이제 밥이 얘기한 것 중에 하나 또 중요한 포인트는 헐투 하이 브레스트 캔서 게스트릭만 합쳐도 그거만 합쳐도 $1 빌리언 시장이 넘으니까 아주 보수적으로 예를 들어서 정말로 헐투 로를 뭐 우리가 많이 못 먹는다 하더라도 헐투 하이만 해도 충분히 큰 시장이기 때문에 우리 헐투가 충분히 뭐 경쟁력이 있을 것이다. 근데 그건 아주 보수적인 에스티메이션이고, 그러니까 헐투 로에서도 지금 효능이 나오고 있기 때문에 헐투 로까지 진출이 가능할 것입니다.
예, 발표 시간상 질문 하나만 더 보고 가겠습니다. 질문.
Due to time constraint, we are going to take the last question because time is running out.
뭐 특정 ADC랑 비교하는 거라서 뭐 저희가 정말로 풀 데이터셋을 이렇게 쫙
This is
테이블화해서
compared to specific
정말로
ADCs. So, for example, we can present a full table showing what would be the safety and toxicity. Without that full table, it would be very difficult to answer this question. So it looks similar, but what are the advantages? So we really need to have very sophisticated data.
the clinical outcome data set from this the another HER2 ADC, it's probably not too easy to answer this question, but you can always add your comments on it.
Well, one of the things I would say is we currently wouldn't consider ADCs with topo-1 isomerase payloads as competitors for the treatment space that we're aiming for. So neither of these, I think both of these ADCs that are mentioned in the question have topo-1 inhibitors, and they will likely suffer in the post and HER2 setting when that becomes relevant in China.
That's a great point. Yeah.
Where and that was part of the concept of developing another HER2 ADC with using the LCB technology, but with a potent tubulin inhibitor like MMAF. Our target market, our competitors are those with non-topo-1 inhibitor payloads. And right now, the clinical data from Fosun and emerging from Iksuda suggests this IXO14 has the potential for best-in-class in non-topoisomerase payload ADCs.
네, 지금 밥에 포스트 NR2 환자를 만날 텐데 여태까지 다른 임상에서 보듯이 토포아이를 맞은 사실은 환자들이 들어왔을 때 또 다른 토포아이 ADC가 효능을 보일 가능성은 사실은 매우 낮기 때문입니다. 그래서 이거를 제가 부연해서 설명드리면 이런 헤드투헤드 정말로 굉장히 큰 환자군에서의 데이터셋은 정말로 둘 다 허가를 받았으면 의미가 있는데, 뭐 달리 말씀드리면 저희 헐투와 이 헐투 ADC가 그런 포스트 마케팅에 비교가 될 가능성이 거의 없어요. 왜냐하면 거기까지 가기 전에 저희가 이 ADC건 저희 ADC건 임상을 2상을 3상을 거듭하면서 저희가 만나게 될 환자들은 first line이 아니기 때문에 이미 키모도 맞고 NR2도 맞고 사실은 이런 환자들을 맞닥뜨리게 되고 그 환자들을 대상으로 저희가 ORR 반응성이나 독성 같은 것을 전부 다 이제 계산해서 나오기 때문에 또 다른 토포아이 ADC가 들어가서 그런 포스트 NR2 환자를 만나서 굉장히 좋은 데이터가 나올 가능성은 사실은 높지는 않습니다. 그러니까 다른 캔서 같은 경우는 예를 들어서 CRC 같은 경우는 뭐 예를 들어 보면은 ADC가 많이 안 들어가 있으니까 ADC를 맞은 환자가 있을 가능성이 매우 낮아요. 근데 브레스트 캔서 같은 걸 생각을 해보면은 요즘 당연히 사실은 NR2나 TDM1이나 헐2 기반으로 한 ADC에 대해서 하는 경우가 많으니까 현실적으로 그렇게 될 것 같습니다.
예, 이번 시간 Q&A 세션은 마치도록 하고.
This concludes Q&A session with Dr. Bob Lutz. And then today there was a media that saying that there will be a clinical trial expansion about how to post. And some people say that the QR code is wrong, so we have corrected the QR code. You can find it outside. Thank you.
Now, the last presentation by Dr. Chingmei Shi. She's going to make a presentation about CS5001, Global Clinical Trial Progress.
Presentation today. I will now introduce you to the audience in Korean. 예, 시 박사님은 시스톤 파마스티컬의 치프
Dr.
메디컬
C
오피서.
is a chief medical officer of Stone Pharmaceutical. She brings over 20 years of experience in the clinical and pharmaceutical industries with deep expertise in oncology and hematology. Let us now begin the presentation.
Thank you. Let me share my presentation slide. Can you see my slide deck?
Not yet. Hold on a second, please.
Sorry.
We are working on it. Please hold on a second, please.
Can you see it now?
Not yet.
Okay.
We are working on it. Please hold on a second. Dr. Shi.
Sure.
Okay. 잘못 올린 것인가? 잘못 올린 거예요? 우리가 올릴 거예요? 우리가 올릴 거야? Everything's okay now. You can start.
Okay, thank you. Good morning, everyone. My name is Chingmei Shi. I'm the Chief Medical Officer from Stone Pharmaceuticals. We are a China biotech company. It's my pleasure today to have the opportunity to introduce CS5001, Stone's global competitive ROR1 targeting antibody drug conjugate. With best-in-class potential and rapid clinical development program, we believe CS501 is a cornerstone of our innovative pipeline 2.0 strategy. Let's dive into why this asset is positioned to capture significant value in oncology and in Stone. A brief introduction about Systone, which is our company. Systone Pharmaceuticals is an innovative biotech company driven by global recognized research and development capabilities. We have established an efficient and high-quality clinical development engine. As of 2025, the company boasts 16 NDA approvals with over 50 data presentation publications and over 10 ongoing discovery projects, alongside over 40 IND approvals. While leveraging a clinical insight-driven modular research and development model, we have a proven track record in high-quality drug development worldwide, with strategic partnership such as collaboration with MNCs like Pfizer to accelerate our commercialization process. Currently, we have 4 commercialized products covering 9 indications across 5 territories, with sugemalimab, which is an anti-PD-L1 antibody approved in China, EU, and UK, demonstrating our strong global momentum. Our strategy centers on 3 pillars. First is a commercial stage program, as I mentioned earlier. Includes sugemalimab, a PD-L1 antibody, and pralsetinib, which are RET inhibitor, and avapritinib, which is a KIT PDGFR alpha inhibitor. And second pillar is our pipeline 2.0, which target high potential novel therapies, including CS501, a ROR1 ADC. This ADC is a global number 2 contender with best-in-class potential, and PD-1, VEGF, and CTLA-4 trispecific antibody. And the third pillar is our early strategy innovations, aims to secure long-term leadership. This includes bispecific antibodies and next-generation ADCs. CS501 anchors our ADC portfolio. This is a compound with global rights, excluding South Korea, and first-to-market potential. This compound targets both solid tumor and hematological malignancies. Currently, we have phase 1b study ongoing, with registration pathways accelerating. Let's dive into CX501 development program. First, let's look into the design of the compound. CS501 is not just another ADC. It's engineered for superiority through 4 key innovations. First is a full human anti-ROR1 antibody for minimal immunogenicity, with DAR to homogeneity while site-specific conjugation. In addition, it has tumor-selective linker, which is stable in serum, but cleavaged by tumor enzyme beta-glucan. And the payload is a PBD dimer prodrug, which can be activated only in tumors. This delivers high potency and low systemic toxicities, comparing with other MMAE or DXd-based ADCs. PBD has stronger killing of slow-growing tumors while DNA cross-linker mechanisms and reduced likelihood of inducing tumor resistance. So why we target ROR1? As we know, ROR1 is overexpressed in solid tumors like triple-negative breast cancer, like lung cancer, ovarian, etc., as well as hematological malignancies like diffuse large B-cell lymphoma like CLL, etc. But the expression is almost absent in most healthy tissues. And Systone's proprietary MAB antibody has validated ROR1's expression via immunohistochemistry, as illustrated in the figure on the right-hand side. In the solid tumors, the expression is around 50 to 90%. Let's look at the preclinical data. CX-501 design translates to exceptional efficacy in preclinical studies. As shown in this figure, CX-501 exhibits high affinity to human binding to selectively to RR1, but not RR2. In tumor cells, on the right-hand side, it shows in tumor cells, CX-501 demonstrates rapid and deep internalization at 37 degrees Celsius. This facilitates toxin release and action. CX-501 also shows potent ROR1-dependent cytotoxicity in solid tumor and hematological malignancy cell lines. For example, in JAK1 cells, which is a mantle cell lymphoma cell line, CX-501 has an IC50 of around 1 nanomole versus around 100 nanomole for ISO ADC, representing a 90-fold activity difference. In tumor xenograft models, a single dose of CX-501 at 1 milligram per kilogram achieved 109% tumor growth inhibition, with 2 out of 8 complete responses. This outperforms the benchmark ADC, even at higher dose level. CX-501 demonstrates robust bystander killing in co-culture assay with ROR1 positive and ROR1 negative cells, eliminating tumor cells adjacent to ROR1 expressing cells. This is important considering the heterogeneity feature of tumor tissues. In patient-derived xenograft models of solid tumors, CX-501 achieved 104% tumor growth index, tumor growth inhibition in ROR1 high tumors, compared to 53% for ISO ADC. This validated CX-501's efficacy in real-world tumor context. I want to share some of the clinical data with you. CX-501's clinical development targets both lymphomas and solid tumors and spans first line as well as later line settings as monotherapy and in combination. Phase 1A is a monotherapy dose escalation study, while Phase 1B is a dose expansion study with pivotal extension potential. As for lymphomas, we target in diffusive large B-cell lymphoma. This includes later line monotherapy. This includes monotherapy in relapse and refractory setting, first line in combination with R-CHOP, second or third line in combination with R-GemOx and R2. In addition to DLBCL, we are also exploring indolent lymphomas such as follicular lymphoma, marginal zone, CLL, etc. As for solid tumor, we are exploring CX-501 as monotherapy in RR1 positive solid tumors and in combination with sugammalimab, which is our anti-PD-L1 antibody in all common solid tumors. Phase 1A data validate CX-501's promise. Data disclosed at ASH 2024 showed CX-501 is well tolerated in heavily pretreated B-cell lymphoma patients. As data cutoff for September 2024, we have 33 advanced B-cell lymphoma patients treated across 6 dose levels, starting from 33.5 to 156 microgram per kilogram. Of the 33 patients treated, more than 2/3 of patients have performance status of 1, and 82% of patients received at least 3 lines of prior treatments regimens. 12% of patients received prior CAR T-cell therapy, and 9% patients received prior transplant. For safety, we have tested 10 dose levels up to 195 microgram per kilogram. We did not identify any DLTs in this heavily pretreated patients group. Safety profile is overall manageable. We have around 46% of patients with grade 3 or higher treatment-related adverse events, with most common ones being hematological toxicities such as anemia, white blood cell count decrease, and liver toxicity such as AST increase. We didn't identify unmanageable neurotoxicities unlike MMA competitors. Recommended phase 2 dose is currently tentatively set as 125 microgram per kilogram for lymphoma, which is dose level 8. In the dose escalation phase, we have observed preliminary efficacy in Hodgkin's lymphoma. Among 10 available patients at dose level 5 to 9, overall response rate was 60%, with 100% ORR at dose level 8, which is our tentative recommended phase 2 dose. This includes 2 CRs and 1 PR. As for non-Hodgkin's lymphoma, among 16 available patients at dose level 7 to 9, overall response rate was 56%, with 70% ORR at dose level 8. This includes responses in both aggressive B-cell lymphoma such as diffuse large B-cell, mantle cell, and high-grade B-cell lymphomas, as well as indolent lymphomas such as marginal zone and follicular.
Dr. Shi, sorry for
data
interrupting.
suggest-
Dr. Shi, so we got the
Yes?
yes, sound is a little winging. So could your mic little away from your mouth, please?
You mean my voice is too weak?
Yeah, it is okay. It is okay now.
Okay, thank you. So let me continue. So preliminary efficacy data suggest CX-501 monotherapy demonstrate a higher response rate than our competitor, which is CLOV, in aggressive lymphomas, over 50% versus around 30% in diffuse large B-cell. And we also, with CX-501, shows activity in indolent lymphoma also. As for solid tumor, CX-501 demonstrates anti-tumor activity in solid tumor with partial responses and stable diseases observed in non-small cell lung cancer, pancreatic, triple-negative B-cell lymphoma, triple-negative breast cancer, and ovarian cancer at higher doses. Notable result includes one partial response in non-small cell and pancreatic cancer respectively, and the stable diseases in lung cancer, in breast cancer, as well as ovarian cancer. This highlights CX-501's potential beyond lymphomas. As for PK profile, CX-501 exhibits dose proportional drug exposure with half-life around 5 days. There's no significant accumulation of free toxin detected. This confirms the linker stability in the serum. Let's move to the commercial potential of CX-501. As we know, CX-501 stands as one of the 2 leading ROR1 ADCs globally, competing with Merck's Zilowski. The key commercial advantages include of course, the first is clinical differentiation. For diffuse large B-cell lymphoma, CX-501 has overall response rate of around 50%, while at tentative recommended phase 2 dose response is around 70%, while the Zilowski has shown around 30% of response rate. In addition, we have broader indications, including indolent lymphoma, as well as solid tumors. As for safety, as I mentioned, we don't have any DLT, dose limiting toxicity reported up to dose level 10. And at ASH 2024, actually Merck presented data for Zilowski in combination with R-CHP as first-line treatment for patients with diffuse large B-cell lymphoma. And utilizing a reduced dose of Zilowski, the combination regimen demonstrates a near 100% durable complete response rate, which is quite encouraging for ROR1 targeted therapy. In addition, Lonca, which is a CD19 PBD ADC, data also supports development of combination therapy with PBD-based ADCs. As shown in this slide, Lonca in combination with rituximab in relapse and refractory DLBCL achieved ORR of 80% and a complete response rate of 50% with no new safety signals. In addition, Lonca in combination with glofitamab, which is a CD20, CD3 bispecific T-cell engager, demonstrates around 100% overall response rate and 78% of CR rate with a well-tolerated safety profile. So these 2 competitors' data actually give us quite promising development strategies for us to move to first-line and later-line combination treatment in diffuse large B-cell lymphomas. As for the global hematologic malignancy market, this is expected to reach $139 billion by 2029, with diffuse large B-cell lymphoma representing the largest indication, which is around 31% of incidence. And solid tumor markets as parallel value, given CX-501's activities in solid tumor like non-small cell lung cancer and pancreatic cancers. And building on CX-501's demonstrated activity and emerging competitor data in lymphoma, such as diffuse large B-cell lymphoma, we are advancing our targeted registration strategies. For phase 1b expansion, we are actually evaluating the monotherapy in relapse refractory diffuse large B-cell lymphomas with a potential of pivotal single-arm expansion. This has already been initiated. And for combination studies, for first line, well, of course, we are evaluating the for first-line diffuse large B-cell lymphoma, we are evaluating the CX-501 plus R-CHOP. And for second, third line, we are evaluating CX-501 plus R-GemOx or R2. So in summary, CX-501 represents a best-in-class potential while optimized ADC design, cleavable linker, non-cleavable linker in the serum, as well as a product design. We have higher response rate in aggressive lymphoma as well as in indolent lymphomas. And this is the first ROR1 ADC has shown promising anti-tumor activity in solid tumors. CX-501 demonstrated a favorable safety profile, which we believe will enable our combination strategy. And currently, as I mentioned, phase 1B study is ongoing for dose optimization for monotherapy in RNR setting and with potential single-arm registration. And the first line and second, third line DLBCL, combination DLBCL, and mono and combination therapy other B-cell lymphomas, as well as solid tumor expansions. We are actually in the process of finding global partnership to optimize CX-501's value. We believe its dual hematologic and solid tumor promise. CX-501 is positioned to transform outcomes for patients and will drive significant value for this compound as well as for System and LigaChem. With this, and this ends my presentation. Thank you.
Yeah, Dr. Shi, thank you for your great talk. Join and lead. 발표가 나온 질문 한번 살펴보겠습니다. 첫 번째 질문
Let's
올려주십시오.
read the first question. Would you please load the first question on the
Hey, Dr.
screen?
Shi, thank you so much for your exciting talk today again. And first question for you just came up. Can you see this?
Oh, I can't, sorry.
Okay, probably it's just coming up. It just takes a couple of seconds usually.
Oh, it's very small. I'm sorry.
The font size is too small to read.
Yes.
Okay. 크게 할 수 있나?
좀 크게 해주시고 그 사이에 한번 말씀해 주시는 게 나을 것 같습니다.
Okay. So I think I can read this for you just quickly. So the question is, in the phase 1B cohort planning, the first line DLBCL trial design includes the combination of R-CHOP with ROR1 ADC. Since R-CHOP has the cyclophosphamide and PBD, so they have the overlapping mechanism. So the concern here is that maybe there would be a toxicity and resistance. So can you comment on the component of the R-CHOP and PBD? Potentially they have the overlapping, the mode of action? Yeah.
Sure. Thank you. Thank you for your question. This is a very good question. Actually, as we designed the first line combination therapy strategy, this is one of the key factors for us to consider. So we choose to have CX-501 in combination without replacing any drug in R-CHOPs for the following reasons. As we know that oncowin, which is a microtubule inhibitor, contributes less critically to the R-CHOP's efficacy than cyclophosphamide and doxorubicin. It has, as we know, it has neurotoxicities, especially for the grade 2 or higher neurotoxicities, quite clinically challenging to manage. Actually, this risk is amplified with neurotoxic payloads such as MMAE, which is used in zero-V design. So this prompts Merck to replace Oncovin in their zero-V R-chip regimen, which apparently they remove Oncovin. And for cyclophosphamide, this remains an essential. We believe this is an essential drug for DLBCL's treatment efficacy, and its primary toxicity is unlike the Oncovin, which is neurotoxicity. The primary toxicity for cyclophosphamide is actually hematological toxicities, which we believe is more readily to be monitored and managed, such as we use the prior prophylactic growth factors, as well as those modifications than persistent neurotoxicity. So we believe that using a reduced dose of CX-501, which we will employ in our combination studies, the combination study, the dose of CX-501 will be lower than the monotherapy. We believe using this reduced dose, this can minimize the risk of overlapping hematological toxicities with cyclophosphamide. So by using this, we anticipate enhanced efficacy of CX-501 plus R-CHOP with manageable safety profiles supported by proactive safety monitoring and dose optimization.
Great. Thank you so much. 사실은 이게 약간의 테크니컬한 퀘스천이라서 조금은
This is a
쉽게
bit technical
풀어서
question,
말씀을
so let
드리겠습니다.
me easily explain the answer to you. In the BLBC hematological cancer, we use a cocktail including R-CHOP. So one of them is cyclophosphamide. You don't have to remember this. It's quite similar to the PBD in our ADC. It actually targets DNA. It also targets DNA. So if two drugs target DNA, it might increase toxicity. There was a concern about that. So Dr. Shi explained another component of R-CHOP causes neurotoxicity. Merck uses MMA2 payload that actually causes neurotoxicity. So Merck could not use all of the R-CHOP. So from the cocktail, they had to remove it. But with our drug, we can use everything. Cyclophosphamide has a bit different MOA, but what's more important is this drug is not neurotoxicity. We can actually manage this toxicity because it is hematotoxicity. So our ADC shows very low hematotoxicity. So if you use a cocktail, toxicity might amplify, but our drug has very low hematotoxicity, and our ADC doesn't generate much hematotoxicity. So even if we add our ADC in the cocktail, it's not going to cause high toxicity. That was the rationale behind our explanation. Let's move on to the second question. Let's read the second question now.
Yes,
Okay.
sure. So thank you. Another good question. Yes, Merck's Zillow has already entered a phase three study. We are, although we are the global number 2 in the clinical development stage, considering the phases of the studies we are in, but we don't believe that we do have other advantages considering our profile. So for Zillow, as I mentioned in my presentation, it has around 30% of overall response rate in relapse and refractory diffusive large B-cell lymphoma as monotherapy. As we know, with this level of response rate, a single arm registration study is challenging globally. So for CS501, actually as monotherapy, it has demonstrated an overall response rate of around 50%, with a higher response rate in the tentative recommended phase 2 dose. With this response rate, we believe a single arm registration study is still possible in China as well as globally. So if currently we have initiated a phase 1B dose optimization study in this setting with a potential of a single arm registration. So if this pathway is feasible or workable, we believe we can register this drug with a much shorter timeline, comparing with a lengthy and costly global phase 3 study as initiated by Merck. So in addition, as we know that Merck's Zillow does not show efficacy activity in indolent lymphomas like follicular, like MZ, marginal zone, etc., and CLL. I think this is mainly because of the payload they use, which is MMAE, which is a tubulin inhibitor. As we know, tubulin inhibitor generally does not work in slow-growing malignancies like FL and CLL, etc. And for us, we do have shown some efficacy signal in the indolent lymphomas, which we believe put us in a development advantage position. So of course we are actively exploring the follicular as well as CLL, etc., which we hope to see some more efficacy signals. And we also have some efficacy signals in solid tumors, which is, we believe CS501 is the first R1 ADC that has shown some signal in the solid tumor. This is a broader stage or broader indication than the lymphomas. And for Merck's phase 3 study, actually it's just initiated, and with over 1,000 patient plans and expected primary completion date of 2029, which is I believe is a long way to go. Currently, we are exploring the first-line combination with R-CHOP and second, third-line combination with other standard of cares. I think with the if we identify or we detect some efficacy signal and prove the safety profile is favorable, we will initiate a phase 3 study very soon. We actually quite confident on CStone's development capability, especially to run the trials. So even for phase 3 studies, we believe we are not late. We still have time to catch up. Thank you.
Great answer. Thank you. 뭐 질문은 비교적 심플합니다. 그 Merck ROR1
The question is very simple. Merck's ROR1 ADC is in phase 3 clinical trial, but this company joins later, but still it's going to be successful. Her answer was very clear. To summarize her answer, its efficacy is really great, and side effects will be lower, so it's very competitive, especially in DLBC, where ROR1 targets. When you use the combination therapies, this is my idea I'm adding to her answer. R-CHOP, when it comes to R-CHOP, you see 90%, 100%. You might think it's really amazing, but naive patients show about 80% response rate. So, but because of that, if you have a high ORR with the combination, it's not really amazing. But when it comes to refractory patients, you need to increase ORR. That's the key. So I'm giving this background to explain the key points of her answer. For refractory patients, when you use monotherapy, Merck's drugs ORR is only 30%, but our data shows 50%, sometimes even up to 70%. So to get approved, single arm monotherapy should be proved to FDA on top of combination therapy. So in their area already we are winning. Not just a hematologic cancer, there's a slow growing cancer like follicular lymphoma, CLL, marginal lymphoma. So for these slow growing hematological cancer, Merck's MME doesn't work really. Microtubule inhibitor works really well for fast growing cancer because they are targeting microtubules for cell growth. But our drug targets DNA, so it works really well for slow-growing cancers. So we can actually expand our indication to hematological cancers. Merck actually gave up on this area, but for the first time, we are using ROR1 in solid cancer, for example, lung cancer and pancreatic cancer. So this is the very first ROR1 cancer therapy that shows efficacy for solid tumors. Because there was no precedence with PBD, because to be effective in solid tumors, toxicity should be low. At the same time, toxic level should be high. But because of the toxicity, they couldn't do that. But our drug has low toxicity. It can be really effective for solid cancer as well. Fourth point, it's speedy. Merck is in phase 3 clinical trial. It seems like they are ahead of us, but their completion is scheduled to be by 2029, so it's a long way to go. So with R-CHOP combination, we can register studies and we can enter phase 3. We can fastly catch up with them. So by 2000, before actually their completion, we can actually speedily catch up with them. That was the gist of our answer. This concludes the Q&A session. This concludes the morning session. The afternoon session starts at 1:20. 5 minutes before the start of the afternoon session, please be seated. Online live stream will resume at 1:20. This concludes the morning session. Thank you very much. So we are going to hand out lunchbox for you. So all the presentation materials for the morning session will be downloadable. You can go to our homepage. So by scanning our QR code, you can access those presentation slides. And the press corps is asking for pictures. So do you need our executives' photos? If as long as you let us know, we'll allow you to take photos of our executives. Thank you very much. Enjoy your lunch.
박 기사님. 예, 오후 세션인 두 번째 파트 넥스트 드라이버스
Ladies and gentlemen, we are now going to begin the second session this afternoon under the topic of next drivers in ADC and immuno-oncology. And the first project will be delivered on highlights of LCB's emerging ADC pipelines. And this first presentation will be given by Dr. Cholong Jung, Head of R&D at LCB. Good afternoon, everyone. I'm Jung Chorung. Unlike this morning, all the presentations will be delivered in Korean. So hopefully communication will be better. So many technologies have been explained this morning. So I'd like to talk about a brief introduction of technology, as well as target selection and payload selection, and what kind of ADC products we are having in the pipeline. So I think that you have a good understanding on the concept of ADC. So ADC is antibody and drug conjugates. And our technology contains linker technologies and conjugate technologies, which is the foundation for our technologies. And then our technologies and product have advanced, and now our focus has been expanded to include antibodies. So you have heard of the excellence of linker system. So the first generation of ADC technologies, as shown in Seattle Genetics and Immunogen. So there is a mixture of one antibody and different types of DR. So we want to improve heterogeneous property. And sometimes the efficacy is low, and sometimes there is a problem with toxicity. So we developed this product, and we went through clinical trials. So we wanted to improve the efficacy and toxicity with our linker payload product. And ADC need to deliver the drugs, and that should be internalized in the cancer cells. So there is a pre-payload in the cancer cells, and then DER is being lowered significantly. So our purpose is to deliver payload to the cancer cells, and this function of ADC is not working properly, and then the antibody has loosened toxicity. That's why it did not have the intended effect. So we need to improve conjugation problem so that we are able to toxicity and tolerability issue. That is the background behind our product development. And if we look at approved tox profiles, it proves that our assumptions are correct. Here you can find 4 ADCs which have already been approved. And all of these have different targets. They are the targeted therapy antibodies. But as you can see here, all of them have same numbers. That is not based on target. All of them are using same MMAA and same linkers. That's why toxicity profiles are same. If the linker is the cause, then if we make changes to the linker, toxicity profile should be different. So that we apply technology making changes to toxicity. And toxicity profile has improved regardless of target. And the monkey toxicity was not target dependent. That was based on platform. That was based on the linker payload platform. So when we analyze other ADC data, for example, there is no improvement in the MED. That is because they are driven by platform. So our linker technologies developed by our company can implement original ADC. Why we are having this kind of toxicity profiles with linker technologies? This data was presented already in the morning. So our ADC is stable, but look at the competitors' products, then 3 out of 4 are put into cells, cancer cells, because these materials are causing toxicities. But ADC has only 1 out of 4. So antibody cannot be effective into the cancer cells. So these drugs onto the antibodies are not good enough. And if that is same antibody and same payload and same linker, if ADC, our link is connected, it is much more effective. And our competitor is using 4 MPK, but ours is 1 MPK, but competitors is less effective than ours. So it is the linker payload makes a real difference. But as you saw in the diagram, 1 out of 4 is lost in the traditional ADC. That is the difference. So through this kind of preclinical trials result, we secure differentiated data, and then we are confident that we can bring benefits through patients based on our platform. That's why we are planning to expand our pipeline. We are not just relying on the linker technologies in order to expand our pipeline. As mentioned by our CEO, unlike other companies, so we are GMP ready and clinical trials ready with a variety of payloads. We are not randomly following. We are thinking about optimal combinations depending on target indications with less toxicity. So we are evaluating on-target and off-target toxicities in order to identify optimal combinations. So also the immune modulators will be developed based on the antibodies. These days, many companies, including ours, are developing bispecific antibodies in order to be cancer selective and in order to have more efficient drug deliveries. So we are working on antibody engineering. We are trying our best, like by investing lots of time in order to achieve optimal combinations. So including this kind of pipelines, preclinical trials, as well as discovery projects are also being worked on. When we select various programs, what is very important in selecting targets? There are many different considerations because we work on a variety of projects. So there are the existing projects and new projects. We'd like to make a differentiation. So as you can see here on this cancer, in the case of lung cancer or breast cancer, so there are known targets and there would be new targets. We want to see whether they are dependent or independent or exclusive with each other. Different expression is not always good. So we want to identify patient groups with medical unmet needs. So we look at target expressions and the mutations. So we can refer to known databases. So there would be validated targets through known databases. And also we can utilize spatial transcriptomics to see whether genes are exclusive in patients or whether they exist in different cells or in the neighboring cells. Through that kind of analysis, so we can make decision how to develop ADC, whether it would be better to be bispecific antibodies or not. And also we consider what kind of combinations would be most efficient for the patients with minimal toxicity. So we do the analysis at a protein level as well as at a combination level. So based on the organ level indications, we are also preparing for backup pipelines. So due to time constraint, I cannot show you all of the targets today. So this morning, as mentioned by Mr. CEO, by the end of 2027, about 20 IND applications will be submitted, which is our goal. So apart from the existing clinical trials projects, many projects are being planned for IND on an annual basis. For example, Cloud 18 was presented, and today CEACAM5 will be introduced, and also STING antagonist will be introduced by Chang. As for the CEACAM5, so we are targeting best-in-class for the solid cancer, solid tumors. That is exclusive to TROP2. So that we are targeting patients with unmet medical needs. So our target is lung cancer and the colorectal cancer. So there will be more elaborate explanation on targets by the next presenter. Normal expression is pretty limited. So that is very good ADC target. and also reverse correlation with TROP2. That is observed very well in NSCLC, meaning that there would be high demand in the medical field.
So we have introduced differentiated antibodies based on the research by Professor Jo Byung-chul. Just like what we have shown in other links and payload technologies, and we are trying to achieve best in class. Then what kind of payloads are the best? TOPA inhibitor, micro inhibitors were used, and we are testing many different efficacies in many different models, and we have actually selected them. This article is well known, and the competitor drug is like U6MP. In our trial, it's a topo drug, and its amount is 3 millimeter. So at the same dosage, the efficacy is better for our drug. So at a lower amount, we show this much efficacy. That means it has really great synergistic effects. So we can actually combine these drugs to produce a best-in-class ADC. So regarding targets, later the speakers will give you more details. Now let me talk about next drivers. The bispecific ADCs. We actually have CD22 and CD20. This is a BCL and BSNHL, and it is targeting them. So we are targeting these patients. CD22 and CD20 are very well-known targets. For CD22, you already have Besponsa in the market. And for CD20, the well-known drug is the rituximab. It's already approved, and it is used as a basic regimen for R-CHOP. So we are trying to expand our ADCs in this type of diseases. As you see here, CAR-T is gaining a lot of popularity and a lot of trials going on that. CD19 CAR-T achieved a lot of clinical success. So CD19 reduced and CD22 is increased. So according to many articles, we see that kind of outcome. So existing drugs couldn't be really effective for certain patients. So for these patients, we decided to develop bispecific antibody for CD20 and CD22. So we are the first company who is targeting these two. So we are going to be the first to enter the clinical trial with that, and also we will be the best in class. We have great technologies, we have great platforms, so we could just choose one of them. But why we are trying to achieve more difficult one by specific ADCs? So their expressions are not consistent depending on patients. For certain patients, CD20 and CD22 are disproportionately expressed. sometimes certain cells are more expressed than the other. So you could develop the monoclone antibody ADC, but there must be a patient with residual disease. That's why we are targeting bispecific antibodies. Its targeted indication is non-Hodgkin lymphoma and B-cell lymphoma. And DIB cell is also one of the targets. As I said earlier, the patients are showing heterogeneous expressions. Look at this graph. This graph is about only one patient. So you see the B-cell lymphoma, and we analyzed with PACs with CD1, CD20, and CD22. So on the left side, you only see the CD22. In the middle, you see both CD22 and CD20 are expressed. And on the right side, you see only CD22 are expressed. So basically, patients show this type of characteristics. That's why we came up with an idea of targeting the two. So only CD22 has developed ADC. So there's no ADC for CD20. So we believe our answer is bispecific ADCs. So we believe the bispecific ADC will be more effective than CD20 ADC or CD22 ADC. When it comes to B-cell lymphoma, we saw how effective it will be compared to the combination therapy. At least we thought it will be the same or equivalent. So when we administered a single time, and it was really effective. When we did a PACS analysis, there are many different combinations of expressions. So most patients have these characteristics. For these patients, bispecific antibody has great potential for ADC. Some patients show only CD20 expression or CD22 expression alone, but for patients with both CD20 and CD22 expressions together, bispecific antibody could be a great solution. So as I said earlier, so we are currently developing twin ADC, but right now there's a CD22 ADC approved, but given all the indices, I think our drug will be the best in class. So our platform is the only one that shows really great PBD-based safety profile. So I think in terms of TI, we will be the best in class. So we are developing not only linkers but also payloads. As was mentioned by the previous two speakers, recently, topo inhibitor, there's a great aspirations for a topo inhibitor and pipeline explosions. There's an increasing number of pipelines for topoisomerase inhibitor. So many ADCs with a topo inhibitor will be approved or have already been approved. So which means that patients will be more exposed to TOPA inhibitor-based ADCs, and it will actually increase the resistance of patients to TOPA inhibitors. So there's a great potential for non-TOPA inhibitor ADC. So we need to prepare a new payload with new mode of action. So we are conducting research on that. So in order to overcome resistance, what type of mode of action is required? What kind of drug it will be? We cannot come to conclusion quick because it will be differ by patient. So we are researching new payloads a lot. So we are trying to develop new topoisomerase inhibitor-based drug. So ADC's resistance is involved in the resistance to the drug itself, but also it involves the less delivery of drug. So 4 or 8 should be delivered to the tumor, but only 1 or 2 gets delivered. So by changing the linker system, such resistance could be overcome, or more potent or more potent or new scaffolds can be utilized to overcome resistance. So among the microtubule inhibition mode of action drugs, we are trying to develop best-in-class. So modulating agent-based payloads and new mode of action payloads. We are trying to develop these. We could do this by our efforts alone, but sometimes we find it impossible. So we are trying to utilize many different resources, including partners outside. So through many collaborative projects, we are trying to develop new payloads. So that way we will achieve innovations. So we started with the linker, but in the payloads, we are also pushing forward with innovations. I wish we could show the new topo I related data, but right now we are at the preclinical stage. So we don't want to run the risk of exposing TPP. So hopefully next year we'll be able to show you more detailed data. But briefly, I could say this much. This is not the existing scaffold you see in the market. And we will develop more potent drug which can overcome resistance caused by the existing compounds. And one more thing is that I said we started with linker, but we made a great leap forward, and we are not complacent with the existing linker system we have developed. Therefore, so one of the questions regarding our platform technologies patent, the patent might expire in 10 years or so, then what is our next step? So we are currently working on the next step. We are developing new linker systems. And also we are going to adopt the low DAR strategy. High DAR can actually worsen PK profile because it can increase a non-specific update. So we will transition to low DAR to reduce non-specific uptake, and by adjusting many different linkers, we will try to control hydrophobic properties of drug. So in order to reduce the one specific uptake, we are trying to develop a more stable, more safe, and more effective linker system. In the near future, we'll be able to disclose our data about that. So it's not this year, but maybe in the future. So linkers technologies and new payload technologies that we are developing will continue to improve our patent profile portfolios. That way, we are going to expand our business values. We started with conjugate technology, but we are also working very hard and innovating in the targets and payloads. With this, I'd like to conclude my presentation. Thank you very much. The Q&A session will happen after Professor Chang Suk-young's presentation. Now, I'd like to invite Dr. Jo Byung-chul. He's going to make a presentation titled Revisiting the Dogma of CCAM's Directed ADC. Professor Cho is a professor in oncology at Yonsei University School of Medicine, and also he is a center of the Lung Cancer Center. He is a great authority in the oncology field, and he has led more than 60 clinical trials, and he's played a critical role in lepotrectinib, laserd, amivantamab drugs. Thank you very much. Thank you very much for inviting me to this great occasion.
So in my talk, I'm going to talk about CCAM5. So why the LigaChemBio need to develop ADC targeting CCAM5? That is the topic of my presentation about myself over the past 10 years. I've been working on the ADCs and clinical development and preclinical trials for both local and global pharmaceuticals, and I have learned some lessons. So when I go to conference on the ADC, many people say ADC is not Lego blocks. It's like the antibody linker payload 1 + 1 + 1 form of a Lego block. But if we make it well, it could be 1 + 1 + 1 would be equal to 6 instead of 3. So that is very promising. And I also felt that during clinical trials as well as preclinical trials, and we've done research with various types of ADCs in my research center. And LCB's the platform is very outstanding. That is one of the lessons I've learned with my prior experiences. And the lung cancer TROP2 ADC has been developed a lot. Some companies have achieved a very modest success. However, they were withdrawn from the FDA approval. And for a Chinese company with the same TROP2 ADC, and they got very significant clinical outcomes. Even for the same target, TROP2, even for the same ADC, depending on what kind of platform they are using, they may have different clinical outcomes. So today, I'd like to talk about CCAM5. So I'd like to talk about different approach by LigaChem on this. I'd like to briefly talk about Ta'an. Some of you may know already. We have done many translational research for the past 20 years, serving as a hub for the cancer-related clinical research in Korea. So we have about 20 researchers, and we also working together with about 54 PhD degree researchers, so that we have lots of experiences on different types of drugs. So I have identified those unmet medical needs while I worked on the cancer-related translational clinical research. So we joined hands with LigaChem to develop CEACAM5 targeting antibodies. Antibody, linker, and payload should be combined in a very optimal way so that there can be minimal toxicity with the patient while having maximum efficacies. So there was a great boom initially about ADC, so Ho3DXD and Dot2DXD. Many drugs were developed, out of which some of the drugs became blockbusters, such as NR2 became blockbuster, but other drugs ended up with as a failure. And the common aim would be increasing efficacy while reducing systemic toxicity to enhance therapeutic index. There are a variety of criteria. A tumor to normal ratio should be good, and also target antigen receptor is also important. So there are certain characteristics to be successful. But it is very hard to find the optimal combination of linker and payload. Then why I think that CEACAM5 is a good target? So we are going to refer to public database, of course. But also, as you can see here, we have collaborators. Internally, we have accumulated patient database and annotated clinical samples and single cells and bulk cells for target discovery. So we are using this kind of database to prove that CEACAM5 is great target. We targeted 100 lung cancer patients. This is a single cell RNA sequencing. Please take a look at red dots. That is specific to epithelial cells, especially TKI refractory patients have a high level of expressions. And also, as you can see here, CEACAM5 high patients have lower survival ratio. And also at the protein level, we have about 100 lung cancer patients, and as you can see here, tumor to normal ratio is pretty good. And also at the protein level, TKI refractory patients are pretty upgraded. And not just lung cancer patients, but also colorectal cancer patients have a good normal to tumor ratio. So we created radar plots to prove that these are the great targets, including internalization and efficiencies and other parameters. And we got to know that the CEACAM5 is great ADC target. So this is Sanofi's data. As you can see here, colon adenocarcinoma, stomach adenocarcinoma, and CEACAM5 has a great tumor to normal ratio and specific to tumor epithelial cell with a high expression. There are the various biologic evidences. CEACAM5 can be expressed in epithelial cells, cell polarization, and the cell architecture are also affected. And not just tumor intrinsic, but also tumor extrinsic mechanisms, especially immunism mechanisms, can be modulated. So when we engage that as ADC target, then we can increase therapeutic effects based on a variety of biological data. Why do I really think that CEACAM5 is a great target? So as you can see here, tosamitamab ravintenzin, CEACAM5 ADC of Sanofi. I was the lead author of clinical trials from phase 1 through phase 3. So I was engaged with the CEACAM5 ADC development directly. That's why I know what's good with this and what kind of additional potential we may have. So I like to briefly talk about my own private experiences. So tosamitamab ravintenzin is the Sanofi's developed one that features humanized antibody specific for CEACAM5, and also as for the cytotoxic agent, it used TM4. Looking at phase 1 and phase 2 data, overall population is like a high expressors and moderate expressors. ORR is about 20% in high expressors. That is pretty promising, and not just response rate, but also with a reduced tumor size. Half of the patients have not reached PR, but they have shown tumor shrinkage. Another factor I'd like to mention here is that if you take a look at the Suma Plus, many patients are not showing partial response, but they are remaining with a stable status. So these are the clinical benefits. The key message I'd like to talk about, half of the partial response patients have maintained PR for more than 1 year, meaning that this medication has very durable benefit. Based on promising data of phase 2, phase 3 was conducted. And out of CEACAM5 positive data, so the standard of care was approved in the 1960s. Compared to the standard of the care, clinical trials were conducted. And dual primary endpoints were PFS per IRC and OS. Unfortunately, they did not show significant achievements in terms of dual primary endpoints. But one thing encouraging here is that I told you about durability of this drug. Blue curve represents long-term benefits, which is pretty encouraging. About 20% of the patients shown PR, partial response, and the durability of the patients of PR was 11 months, which is pretty good. So this drug has some limitations, but if we overcome those limitations, it can be successful clinically based on this table. And another limitation we need to overcome is, if you take a look at this data, CEACAM5 tumor expression, if expression is higher, there could be higher benefits in patients. And soluble CEACAM5, if soluble CEACAM5 is high, it may lead to poor performances. So we need to develop antibody which is not bound to soluble CEACAM5. And one thing very interesting out of this data is in terms of adverse events, such as those reduction and discontinuation, that was pretty low. However, those delays because of adverse events were higher relative to docetaxel. I'm going to tell you more later on. That is because of DA4 related toxicity was not recognized at the time of development. That is very horrible ocular toxicity that can be generated to patients. So people may become blind because of this toxicity adverse event. So these are the issues we need to overcome, these limitations. About 30% of the overall patients may have these adverse events. And the patients and the doctors would be scared, which may lead to low the complacency. So I was engaged with the CEACAM5 directed ADC clinical trials. So I think that as long as we overcome these limitations, it can lead to success. For example, because we are talking about refractory patients, so we'd like to improve response rate more. So CEACAM5 is antibody, but we'd like to make it with better internalization. Even for moderate expressors, there is certain level of activities. So affinity should be good. Avidity should also be good, so that we can deal with not just the high expressors, but also moderate expressors. Third one is CEACAM5 plasma shedding. So that may have a negative impact on the patients. So it should not be bound to shedding plasma. The fourth limitation is about ocular toxicity. So usually doctors cannot bear ocular toxicity. So as long as we overcome these limitations, things are pretty promising. So our goal was to develop antibodies with these characteristics. We developed many clones, but recombinant CEACAM5 or high affinity, or the CEACAM5 clones which are not bound to soluble CEACAM5, or with a higher level of internalization, so they can have higher or better activities for high and moderate expressors. So again, that is very important not to be bound by soluble CEACAM5. So we used various patient samples to reduce binding. That was proven by different types of data. So less binding and better internalization. So we compared our antibody against the tuzamitamab for the therapeutically clinical higher doses. Binding is better, or affinity is better, so that compared to tuzamitamab, our antibody has better binding abilities. why we are expected to have better binding or better affinity. We compared antibody characteristics. Tuzamitamab is bound to A3 and B3 at the lower end, but our antibody has binding, the multiple binding abilities to both the higher end and lower end, which lead to enhanced binding capabilities. This is another data. The antibody has dual binding characteristics, so it can have better affinity, and also even through epitope mapping, it shows better binding. Why it has the dual binding, including A3 and B3? If we take a look at sequencing, so the antibodies bound here can also be bounded to the lower part. That's why it can have better affinity. So this kind of characteristics of the antibody can be associated with higher internalization in theory. And as you can see here, one antibody can be bound to ANTOM and A3B3, and when the dosage is higher, then that can also be ended to other lower ends of CEACAM5, which can result in better internalization. We conducted the various in vitro and in vivo experiments. If we take a look at this table, we used LCB platform. Compared to tuzamitamab ravtansine, our antibody has great tumor-to-normal ratio. It is lower in normal cells but great in tumor cells, and in vivo benefit with the same dosage of tuzamitamab ravtansine. The ADC has even greater effects in vivo data. Global development is working on, including M9140, EBC129, and Pfizer. So we have great ADC platform for troubleshooting existing unmet medical needs, and then we like to aim for the first FDA-approved CEACAM5 ADC. Thank you very much. Thank you for your great presentation. So Q&A session will be conducted at the end of Ms. Chang Soo-kyung's presentation. So we did joint research, secured those data sets. So now we'd like to have a Q&A session. So the reason why we did CEACAM5 high was that all the pharma companies want to deal with as many patients as possible. So I told about 20% response for the high and 5% for moderate expressors. But actually, the efficacy of a drug itself is very important, intrinsic efficacy. But we need to consider various combinations of factors such as patient's compliance. In the clinical settings, the eye infection, ocular toxicity is very serious matter we need to consider. So those horrible adverse events, it would be paper talks. But in terms of hands-on management, that is one of the adverse events that is very hard to deal with from the perspective of doctors. So we need to consider high as well as moderate expresses in order to expand indicators. So not just payload differentiation, but also antibody differentiation would also be important. Thank you very much for the answer. Let us move on to the second question. Regarding MMA, that was developed by Singapore's A-Star. So the low incidence of neuropathy. MMA has about 50% of incidence of neuropathy. We use different ADC platform. It does not mean that we can overcome the payload-related toxicities completely. For example, prior researches. For example, actually, the treatment durability matters. Because as for the neuropathy, when the medications are they are administered for a long term, there would be negative impacts. So for patients with shorter treatment durations, there is less incidence of adverse events. That is also related to the MMAE payload-related adverse event. So as long as we can minimize non-specific payload release, then I think that is very manageable when we use ADC platform. I think that is about Merck Sequen-5 ADC. When TOPA is used, then hematology cannot be avoidable. As long as we have that payload, ADC platform can be enhanced with link stability. In this regard, I think that LigaChem Bio's the platform is great. Yeah, that is the answer. So we do not have additional questions. So if you have any question from the floor, please raise your hand. Actually, we have another question pre-registered. So I'd like to ask for your patience. We are currently typing the third question. So your questions from the floor are welcome. What is the main mechanism of ocular toxicity that is related to payload, not related to target? That is because DM4 was utilized. Why DM4? Because that is characteristics of that payload. When I consulted with LigaChem Bio, so I told that payload selection is very important, and the LCB is doing great about that. Any additional questions from the floor? If you have, please raise your hand. If there is none, we'd like to conclude the session with this. Thank you very much.
Now, would like to invite the Center of Immuno Anti-Cancer Center, Dr. Jang Soo-kyung. She's going to present on when LCB stings cancer. merging chemistry, linkers, and immunology. She has over 22 years of experience in immunology, and she is in charge of developing next generation drug. We are learning a lot about ADCs. In this session, I would like to take you to a different landscape. You might be a bit tired and dozy. I would like you to wake up and join me in this new adventure. Let me talk about cancer immunotherapy. The existing chemotherapy and target therapies are directly attacking cancer cells and killing them. But immunotherapy is activating immunocells, and these immune cells are controlling cancers. As you see, there are many different therapies, not only for cancer but also for other diseases. Merck's Keytruda is a PD-1 antibody-based immunotherapy. It's very well known, and it is recording top sales over the last few years. And also this year is going to see the #1 sales in this drug. And also for when you see the drug is one of the key metabolic diseases, and it is a driver disease in the pharmaceutical industry, and its prevalence is increasing, and PD-1 is very good, but its response rate is like 20 to 30%. So we need a drug that can complement the Keytruda or the Porter drug. So we are trying to develop the complementary drugs. Keytruda was approved in 2013, and it has many side effects, and many research has been done. As a result, at clinics, such side effects has been handled better than before. So the tumor microenvironment, so we have more understanding about it. And given this practical situation, immune checkpoints and many different immunotherapies are being developed. For example, STING and cytokine modulators. And these modulators are being attached and combined with antibodies. And also biospecific antibodies are being developed. So there's a great need for breakthrough anti-cancer drugs, and we are actively researching on that. LigaChem, you might be curious about LigaChem developing immunotherapy, why we are doing this, and if we do so, what type of immunotherapy we are developing. You might wonder about this. In a way, LigaChem is developing immunotherapy. It's not a weird thing. Keytruda has been used at clinical settings for decades, and there have been thousands of combination therapy trials. And as a combination therapy, chemo drug is most effective. As you know, ADC is a conjugate attached with chemotherapy. So maybe we can add ADC, then it will be very effective drug. It will create very great synergistic effects. So LigaChem's chemotherapy and chemistry and linker technologies, they can be combined to target IO. So we are developing such a payload like this, and this development strategy is associated with LigaChem's Vision 2030. So in terms of expanding our payloads, so we are developing many different types of payloads. with many different mode of actions. So IO drugs and other MOA payloads and toxin payloads can be conjugated. By doing so, we can create a new modality. So it is also in line with expanding modalities. So ultimately, ADC is very effective, but we want to deliver this great efficacy to as many patients as possible. So we are trying to develop combination therapy with ADC. And also we want to test and prove the efficacy of combination therapies. So it is also in line with our directions of reducing toxicity. So we focus on STING in applying our technologies. STING is a protein that our cells have. STING is attacking outside attackers like viruses or bacteria, or if our immune or cells are sensing this is something foreign body is not normal, that kind of sensing is happened by the STING. So the STING is actually triggering the immune cells to activate immunity. So in terms of cancer, it might be a little bit complicated, but to give you a nutshell explanation, let's say in a cancer cell, there is a abnormal cell, and that information is communicated to our immune system by STING. And STING also activates the immune system, and it also activates immune cells. So if it combines with the anti-cancer drugs, it can actually maximize its effect, anti-cancer effect. It's called STING agonist. So LigaChem is developing STING agonist. So based on its proprietary chemistry and linker technology, we are trying to develop STING agonist in the form of anti-conjugate or small molecule. That way we are trying to secure our competitiveness. I think I skipped one important part. I think I will discuss it later. LCB39 is a program involving STING agonist small molecule. So we are developing a STING agonist a small molecule, and we aim to IND stage next year. STING agonist LCB39 is a systematic administration IV drug, and our highest priority is on safety, and we actually conducted a toxicity test in monkeys, and we secured substance that can actually ensure safety when it's administered. Although LCB39 is a synthetic, but it is a specific characteristic of being accumulated within cancer. So a STING agonist can actually convert that. Let me give you more details about LCB39. LCB39 is a direct form STING agonist. And its LGB's beta glucuronide trigger is attached. It's conjugated like that. So it is increasing the soluble quality, and as it's attached, its penetration into cell has been reduced a lot. So although it's administered systematically, it has very high safety. Although it's safe for the whole system, as I said earlier, LCB39 has a characteristic of being accumulated within the tumor cells. So it's a very rare characteristics, but LCB39 has this property. Additional property is that the enzymes expressed in tumor and so it actually activates LCB39, and also it increases the metabolite. So with this, we can actually catch two birds with one stone. This is the data. Let me briefly explain to you as easy as possible. This is the in vivo efficacy result. In many mouse models, we conducted in vivo efficacy. So it's a dose-dependent, dosage-dependent cancer control effect has been very excellent. And we see the cancer cell doesn't grow with this. So basically, immune memory has been created. This is another important point. This graph might look so complicated, but don't be overwhelmed. Let me easily explain it to you. This model is called ART, lung cancer model. So in the lung, we planted tumor, and then we administered the LCB39, and we see this great result. So we actually extracted a cancer cell, and we actually proved whether LCB39 has the anti-cancer control. So when you look at the red bar, it shows really big difference. So the T cells are much activated, and T cells can be activated, and many T cells can be created by the relevant cells, and the number of those cells has increased a lot. And the cancer cell eating cells have been amplified as well. So we see the result. So LCB39 has increased the environment from cold to hot. This is the data that proves that. This is another differentiating point of LCB39. We really want to highlight this. So we administered LCB39 to the mouse with tumor at 3 different doses, and then we extracted blood and a normal organ cell, and then we wanted to test whether there is the active metabolites or LCD39 are found. So we actually observed that the metabolites and LCD39 were present in those cells. So we conducted further research. So the blue line is the amount of a drug that can actually activate the metabolite. So as I said earlier, metabolites were observed, and the dosage that can actually activate metabolites is the red line. So 1 milligram kg. So we see a lot more amount here, and even at a less dose, we were able to detect it. So the amount of LCD39 is doing its job. So according to this data, we were able to verify that. This is a combination potential. This is a very important part that is specific to our company. We are conducting many different combination trials, particularly ADC and PD-1 combination trial. This is the Dato DHC and Trux ADC 29. So you see the LCB, when LCB39 is administered in combination, as you see in the red line, the result is very homogeneous and the performance is really excellent. As you see in this data, LCB39 can be utilized in combination with the immune checkpoint or the ADC drug, and it can actually deliver more benefits to patients and also offer treatment opportunities to patients. And this kind of potential market is really possible. So you might ask this question. So pharmaceutical companies have been developing drugs using STING agonist, but there's no success case in clinical settings. There exists such a challenge. So in my company, we look at this in two perspectives. First, compound itself. As I said earlier, LCB39 has very differentiating characteristics. Although it's a synthetic compound, it can be selectively accumulate within tumors, and the normal cell toxicity has improved a lot. So such characteristics are being shown by our drug, and it offers really great potential. Another thing is that clinical translation. Existing pharmaceutical companies do not have a special clinical strategies or differentiating strategies. Although it's a great drug and great target, but you need to also have a great clinical strategies. So when you conduct clinical trials, you need to have really great strategies. And what we are also focusing on biomarkers. What kind of patients are really responding to our drug? So we will focus on biomarker studies. Currently, we are actively pursuing biomarker studies. So the clinical designs and with this drug, and we are doing our best to become the best in class and first in class. Now, let me focus more on ADCs. By utilizing STING agonist, let me talk about our anti-conjugate. So you learned a lot about LigaChem's link technologies and the ADCs. You see the immune modulator here. You can actually apply to AIC. So the site-specific characteristic can actually precisely target, and this technology applies to immune modulator as well. And also, I mentioned that we are expanding modalities. So ADIC is one of them. So ADIC has this form of drug. How many toxins does it have? How many immune modulators does it have? So we can actually accurately create these based on our LigaChem core technologies. One example is that STING agonist DAR4 target A drug. This is one example of AIC with those characteristics. So this is the translation model. See, you can see the dose. It's a dose dependent, and it shows really great efficacy. Effective molecules for STING agonist are well expressed, and you can see that in molecule STING agonist AIC. This is target B ADIC with DAR2. So target B is less expressed in this model. The red colors have two different concentrations, and their efficacy is limited. But although with the same DAR, but compared to the same dosage of competitor, it has a better efficacy. And if you have a higher dosage, and we can see the complete remission. So when you look at the STING agonist used in ARC and AIDC, they have a different structure compared to LCB39. So we chose the most optimal payloads, and this is the most optimal STING agonist. The AIC and AIDC, as I mentioned in those areas. So it has the optimal pair and optimal combination, especially ADIC. Within one drug, there is DUI present at the same time, so it creates a synergistic effect. So there's a certain MOA, and when you have two drugs with different MOAs, its potency increases. So in this combination, because they have two different MOAs, so memo function and also the immunity activation, so it has very long-term efficacy. This is the combination of ADC and IO. At LigaChem, not only STING agonist, but also IO and direct T-cell activating payloads are being developed. So there are areas where there's some needs that couldn't be addressed by the existing ADC, so we are developing next generation ADCs. So hopefully our this development strategy will bring about the next generation ADC, which is the best in class. Regarding the presentation by Dr. Jung, Dato ADC and PD-1. So we actually did a Dato ADC because we wanted to show the better efficacy compared to the existing drugs in market. So we are going to open a Q&A session. Dr. Jung and Mr. Jung, would you please come up to the stage? I think it would be better for me to stand up so that I can have a better view of the questions on the screen. So would you please load the first question? DAR 1 and low DAR was mentioned in this question. We increased the therapeutic index. All ADC's therapeutic index is affected by the linker and payload as well as targets. So as we pursue low DAR, we can actually increase number of targets where PBD can apply. So we can actually expand our market in the new area. That's why we are pursuing low DAR. Currently, our drug is safe, but by lowering the DAR level, we can actually increase the safety even further. By doing so, we can apply our drug to the indications where the existing drugs cannot target. Second question. Earlier, I briefly mentioned about this. There's no complete clinical design developed yet, but we are focusing on biomarker, and we are trying to identify the patient groups who show the drug prove our drug activity. So we are trying to develop relevant biomarkers regarding that. So we are going to do the clinical design. On top of that, we are trying to identify the STING agonist efficacy and toxicity. That way we are trying to design our clinical trial. Thank you very much. Third question. We have quite many questions. You are comparing the monoclonal antibody and the biospecific antibody. Although you are targeting the same thing, depending on the epitope or affinity, it could actually develop different toxicity. So although it's a biospecific antibody, what kind of target combination, that matters, and how you can adjust the affinity for each target, and what kind of biospecific antibody platforms you're going to use. Depending on that, its efficacy and toxicity might differ. So we don't just simply developing biospecific antibody. So we need to modulate and adjust the affinity and the number of payloads and what kind of platforms we are going to use. We are looking to many different aspects. So although biospecific antibody has general benefits, we are not just pursuing them, simply looking at those benefits. CMCs might have challenges, so there's more steps involved in that. So it actually increases the production cost. In that respect, the existing biospecific antibody platforms can be utilized. Based on my experience, this is my insight. Next question. This question is quite long. Regarding this, we often receive a question like this. This is about a STING target. STING is activated chronically. It can be actually pro-tumor. So this kind of signal might actually go towards pro-tumor direction. So the question is about that. In a way, even in the academia, there's no clear definition about this. However, when you look into the literature, you can find out the following. Chronic pathways and signalings are activated. Information can happen. So it can serve the role of the stage where the cancer is being created. But most patients we see are suffering from already advanced and aggressive cancer. So they are not at the stage of developing cancer. They are not on the initial stage. STING activation versus drugs. So when you compare the two, they are different. When you look at the non-clinical testing, small amount STING agonist versus a high dose STING agonist, when you look at the cancer reaction, it's all dose dependent. So it is different from the cancer initiation pathway. So the STING agonist therapy at the stage of treating advanced cancer is different. the cytokine release can be strengthened. Although it's the same STING agonist, the amount that releases the cytokine or the amount that does the opposite, they're kind of close. But we found the STING agonists, well, of which window is quite wide. And also dosing schedule period. So we don't do it frequently, just once every week we administer it. So we administer it and then give time for the immunity to be recovered. So I believe we can actually adjust it by spacing and scheduling it properly. So we need to change the way we think. IL-6. I think that the person who raised this question knows a lot about STING. So when STING is activated, the inflammation pathway can be turned on, or there's another pathway, but our drug, we don't know why, but our drug is cytokine release syndrome. So we don't see that CRS pathway, and we see the pinal dominant interferon pathway. We see type interferon pathway more with our drug. Another question? Another question about STING? This is a great question. In the biomarker studies, this area is included in our biomarker study. Do I need to explain more? The existing pharmaceutical companies who developed STING agonist didn't consider this, but beta glucuronidase, that is our specific one, and they didn't consider this part. Biomarker and study designs will incorporate this, which will be differentiated from the existing clinical trials by other companies. So we are not in an opportune time to answer that question. Regarding this is what we are actually doing right now. We conjugate IO payload to antibody. That way we can have more activation in tumor. So we are trying to add more drugs to fight cancer. So you talked about differentiation. I don't get it clearly. So what cells the antibodies are targeting? So that could be a differentiating point. The LCB39 is a chemistry drug, but it can be injected into, it can be delivered to a tumor a lot, and it also has a properties of the antibody conjugate. So our STING agonist could be different in terms of what type of cells it is targeting. And also it's different from the general STING agonist because it is targeting cancer, and it brings more STING agonist to cancers, and it is exposed when it is exposed to the entire body, the systematic toxicity can be reduced. Next question. Mr. Jung can answer this question. Target affinity. I think you're talking about target affinity. With a higher target affinity, that doesn't mean that internalization is being done well. So neutral pH affinity and acidic affinity, depending on that, toxicity and efficacy might vary. And in terms of pharmacokinetics, on rate, off rate might vary depending on whether it is the liquid cancer or solid cancer. So for solid cancer, off rate is more important. And the within tumor cell infiltration rate matters, but the movement into the tumor is also important. So proper balance is important. So cellular infiltration and the tissue infiltration, you need to modulate those rates for optimization. Next question. The existing targets, for example, HER2 and TROP, and they couldn't find the accurate biomarkers, HER2 positive or these things are based on Herceptin and also IHC. So you need to have a different you can actually need to have a different standards. So for novel target, selection of biomarkers is very critical. So when we started developing, we already started selecting biomarkers, but when we select targets, we already start a research on the selection of biomarkers. So if it is a novel target, biomarker is more important. The last question, and then we are going to take a break. Last question. This method you mentioned in your question is really troublesome for us. You need to actually do it, and you need to pursue both depending on indication, although it is the same target. Sometimes ADIC might be more appropriate. For example, ADIC might be more effective for breast cancer. For other disease, ADC might be more effective, and after that, immunomodulation could be given. So you need to take a personalized approach. You don't just choose one. You can actually develop both to a certain degree. Basically, internally, in the past, best in one class ADC for one target, but now we are trying to form best package for the relevant target. So we are trying to form the package drugs for the same disease. So we can actually cover all relevant diseases with the package. Because we are trying to develop such package, it takes a lot of time, and also there's more research projects being underway. Let me give you more explanations with focus on ADIC. ADIC, you might think ADIC might not be different much. The two good drugs can be used in combination, but those two drugs can be loaded onto one modality as one drug, and its advantages are being really clear. So compared to the combination therapy of those two, if you add them onto one modality, it shows some differentiating effect in terms of toxicity and efficacy. So BAC and the STING agonist can be loaded onto one modality, and it creates really great synergistic effect, which is very attractive drug. So in the second part, we talked about new payloads, strategies, and ADC pipelines, and also STING-related discussions. So this concludes the second part. And at 3:10, we are going to start part 3. Thank you very much.
Ladies and gentlemen, we are now going So we'll now begin the final part on the LCB's business strategy. The first presentation will be Global Out Licensing Strategy by Executive Vice President Jae Choi. As Head of Global Business Development, EVP Mr. Choi is responsible for external partnerships, including the global out-licensing of LCB's pipeline assets. He has successfully led licensing deals with major global companies such as J&J and Ono. Hello everyone, my name is Jae Choi for Business Development. So it's been a long day today, and the content would be very challenging to understand. So I'd like to thank your effort and commitment. And you must be very tired after the lunch break, so you can stretch yourself prior to this presentation. It's been a year since I joined the last presentation, and this would be an extended version of last year's presentation. So there are several components I'd like to add more on top of last year's. The topic of my presentation today would be very long or short. Depending on the situation, I'd like to talk about business strategies over the past 1 year and what would be the future landscape or strategies or the goals in terms of global licensing. This morning and this afternoon as well, you have heard of LigaChem's research and development, current trends, and the future plans. And the final outcome of all of these activities include licensing and the business strategies. So the theme of this presentation relates to global out-licensing. What do you think would be the most foundation for licensing? Because we are a technology provider, so that what is most important is technological foundation as a provider. I think most of you agree with what I'm saying. So you guys are the experts in the biopharmaceuticals field. So I'd like to ask you this question. Let's say there is a company saying that they have great technologies. What would be your criteria? Let's say there are companies like A, B, C, and they say their technologies are great, outstanding on the global stage. Every one of them is boasting of their outstanding features like a second generation, and they are leading technologies in certain fields. What should be the objective evaluation criteria or the method? What would be fair evaluation? Have you ever thought about that in terms of evaluation? That might be a difficult question, but there is an easy answer in 3 ways. One, so we will see whether that is recognized by the one of the most credible societies in that field. I think battery is running out. The pointer is not working. Now it is working. Fortunately, ADC Society, World ADC Summit has been held from 2010 every year. That is the most recognized society. And then they are recognizing the best their ADC platform, and the LigaChem has been receiving Best ADC Platform Technology award for 6 years consecutively. And what is the criteria for selection criteria for this award? About 20 to 30 companies are being designated as the candidates, and then there will be first line voting to decide top 5 companies. And then through this society, top 5 companies are being notified of their selection as the finalists. And then there will be expert review committee to go through the voting process. Then there are the scholars from the academia. But the committee is composed of the real experts like a CEO, CSO of global biopharmaceuticals, or the doctors who are really actively engaged with the clinical trials. So the first place is winner, and second place is runner-up. And we have been the winners for 3 consecutive years these days. So not just in ADC. Let's say there are certain words, then like think about how many Korean companies we are having as top tier in this kind of world-recognized society. We have other ADC companies, and what we want as a LigaChemBio is that we'd like to see more the ADC companies so that we can have like a fair competition so that we can contribute to the ADC. That in order to be really recognized as ADC objectively, they need to be ranked as top 5. Number 2 is that clinical results matter. New emerging technologies. Let's say they would have great results in animal testing. However, that may be different in the clinical trials. So, like having proven results from the clinical trials are very important. For example, relative to NR2, like a smaller administration and comparable efficacy. And the neutropenia is about 67%, but ours is less than 10%, which is showing the real differentiation, and that is proven by the clinical trials. So that is the evidence of outstanding technological power. Let us go back to the technology transfer. Have they ever done technology transfer or out licensing? So we did it with Ono last year, and the total value exceed 20 billion right now. So that purchasers are strict evaluators, I like to say. Let's say if you like to buy the one item which is worth only like 100 million won, you are reviewing very strictly, but this is worth about trillion won. So that purchasers matter. They are evaluating very strictly technological advancement and clinical possibility and commercialization. We've gone through various due diligence relating to technology transfer. So when we receive about like 40 to 50 questions, like we are preparing for the answers very thoroughly. But actually, it's like 350 or 400 questions we get for the due diligence, and for every question we answer with about the scores of presentation slides, and that need to be passed before reaching an agreement. The reason why I'm talking about this process is that through technology transfer, due diligence experience really matters. And through that, a company can advance more. So recognition from the society, and then validation through clinical trials, and also technology transfer or the out licensing are the 3 most important criteria, I think, and we have gone through all of these criteria. And I mentioned this last year, market trend cannot be ignored at all. Depending on the market situations and technology transfer, things might differ. And one thing peculiar about last year's market, so if you take a look at the MMA prior to 2022, the total value was about 20 trillion a year, combination of like MMA. And then it went explosive from 2023, exceeding 150 trillion won. That was because of the success of NR2, and also as presented by Bob Roth, then we were seeing growing number of clinical trials and high interest among global pharmaceuticals companies. And with the release of ADC materials, the market revenue was generated significantly. It's been only 1 year since the last year's presentation, and about whether this hot market is being maintained or not would be another question. In 2024 alone, we got more than 10,000 deals, and that trend is continued into 2025. Even considering FX, it's about worth 99 trillion. But the value looks less relative to 2023. However, 2023 was a year of M&A. So M&A value was about 40 to 60 trillion won. So excluding that, in 2024, I think the value is comparable to 2023. And after the last year's presentation, including Ono deal, so we are continuing to have a trillion won worth of deals. So again, we are having years of the M&A, and not just clinical deals, but also preclinical stage deals are also being signed, meaning that the big pharmaceuticals would like to be engaged as early as possible, being engaged with preclinical stages. So in 2025, there were the early stage deals. Known targets and first-in-class targets were having active transfers of technologies. A new trend is growing demand for bispecific ADCs. So there are the early stage deals for bispecifics. So all of these prove that our direction is right. Now I'd like to switch my gear to talk about our models. There are two different types of technology transfers. One is like complete product, including antibody, a linker, and payload. So we do the development or the IND or to some certain state. Or we provide only platforms and all the rights would belong to the buyer, as long as they have antibodies, and then they work on the development for this ADC. For the big pharmas like MZL, they like to get the approval, so they are willing to give the royalties, so there is no possibility of third-party technology transfer. But if royalties are given to big pharma technology providers, and then there can be like a milestone-based revenue again. So as by HER2 and the system, there is a big possibility of big pharma technology transfer that would be another source of revenue and cash flow. So you may have heard of that today. So it was expressed represented as ADC, but we are talking about AIC, ADIC, and ADC today. So regardless of product or a platform, new innovation, new payload, IO compound, AIC. New innovation is already on the way, so that our licensing value would have significant growth once again. That is because there is existing demand on the global stage. So in terms of product, that picture is pretty clear, especially recently. And technology transfer with Ono, you might think that the value would be lower than Johnson & Johnson. We signed confidentiality agreement, of course, but Johnson & Johnson got the technologically valid one, but for Ono got the non-clinically non-clinical stage one, so that the value is pretty high. And platform prices are also rising continuously. And our CEO briefly mentioned about Vision 2030, speaking of which, by 2030, here is our direction of development. The summary is represented by Vision 2030. Before last year, we wanted to have about 2 to 3 candidate materials per year. So given biotechs in Korea or the US having only one candidate material is already good enough. But our aim is to have about 4 to 5 candidate materials per year. That's already been declared. So within 5 years, last year we announced that would be 10 to 15. But our CEO mentioned 20 candidates within the next 3 years. So I'm not really sure whether you took notice of it or not. Because of delay, companies usually trying to reduce the number. They are extending the period while reducing the number of candidates. So this is new, the disclosure according to our speed. So we are pretty much on track, and we are accelerating. And in terms of BD, through new innovation, our technology are having more added values. And another implication from this, like in 3 to 4 years, we are going to have 20 pipelines. We do not need to develop all of this on our own. So as for the global clinical trials, 5 to 6 will be ongoing, and for the remainder, we can have technology transfer. And we often get questions whether we will continue to transfer our technologies. The answer is yes, we will continue to transfer technologies for the single assets and platforms. Last year, I also mentioned that we were going to do package deal platform combined with pipelines, and we lived up to that promise. When you take a look at the owner deal, so non-clinical, the materials were transferred worth about 700 trillion won. But platform deal is totally separate deal. We didn't want to disclose the value of the deal, but again, the deal size is growing more and more. And then it was extra technology transfer. So 2 technology transfers with Ono. So as for the meaning of this, apart from the revenue, there are reasons behind this kind of deals. Speaking about Ono, because of Pembro, Opdiva is hidden, which is competing product. And then this is the original developer, and BES got the technology transfer. And then they sold more than 10 trillion last year alone, meaning that the Ono is receiving royalties from that. And also, it has great experience and know-how in terms of clinical development and commercialization. That's why it has capability of developing SCB-987 in an expedited way. And also, we are selling non-clinical trials at a very good price that is also subject to technology transfer going forward. And when it comes to platform, it has differentiated antibody platform. It can encounter our ADC to produce really competitive ADC programs, and then like milestones and later stage payment would have another possibility. commercialization strategy and global licensing strategies. It was mentioned partly, and I'd like to talk about two new aspects before I conclude my presentation. These are new terminologies. One is BioBest. Second one is Big Alliance Package. Let me talk about them. Let's say there are already proven ADCs. There are more than 20 ADCs which are already approved. But there are limitations even though they have been approved by the authorities. So they are approved, but they are not satisfactory. There might be toxicities, for example, optical toxicities as mentioned by Dr. Zhou, or hematological toxicities, or different efficacy. So 20% would be good enough, not necessarily 40 to 50%. So increasing efficacy and reducing toxicity is the needs we need to address, and this was mentioned a lot in the morning session. Like continuing to use the one payload may have resistance, so that we need to consider refractory patients. So how to address refraction issue? So when it comes to bio-based strategy, I like to rephrase it another way. The antibody should be okay. There is nothing wrong with antibody. Things are determined by platform and payload. So already the released ADC in the market, usually they have no problem with antibody. And those antibodies have already been administered to like tens of thousands patients already. So there is no surprise. That is antibody confirmed in the market. So we like to use the proven antibodies and applying them to the ADC technologies. That's why we name it a bio. Actually, it should have been named as Bio Better, but we are receiving the Best Platform Award from the society, so that we like to pursue Bio Best ADC instead of Bio Better ADC. And also, there are many ADCs that are subject to patent expiration, so that we can make bio-best ADCs from the expired ones. And another thing is fast track. So, like clinical design, what types of cancers. So we need to serve as pioneer to become bio best. So even IND packages are being formalized, so that we can expedite up until the approval stage. And protocols have been accumulated a lot. Here, the red dots represent the patent expirations by 2026 or 2029. So we need to prepare for bio-best prior to the expiration, so that we can work on clinical trials and they could become another blockbuster. As for the antibody, once it is expired, there are a number of biosimilars. But as explained by Bob Lutz this morning, based on the lessons learned, ADC can be advanced continuously based upon the prior experiences. But why not other companies? Why us? That is because we have differentiation through our clinical trials. They need to have technologies to make better ADCs. To elaborate more on this, all of these proven ADC have FDA black box label. If you take a look at the BCMA Black Lab ADC that has great serious apoptox, we do not need to make change to MMAF. We are utilizing same toxins, same antibodies, but our own linkers. But by changing toxins, we can address resistance. For example, neutropenia or hematological toxicity. So the ours has low toxicity, so that we can utilize the same antibody to be combined with same payload. And through the next generation ADC, we can achieve bio best exceeding bio better. That will become great opportunity for us. And already we are working together with big pharmaceuticals on this topic. And then we didn't disclose internal pipelines much so far. But through the bio-based strategy, we are already working on a number of pipelines. Lastly, that will be the ultimate licensing strategy, like a single asset technology transfer worth like a 2 trillion, and then a package deal including the platform. And the future direction of global licensing would be multi-asset platform. I'm not talking about one asset, multi-assets, and then different stages like the one clinical stage and 2 preclinical stages and like 2 research stages. So it will be a strategic alliance or the multi-asset platform package, which is part of our big deal. Of course, deal size would be several times bigger than the existing deal. You might wonder, in my opinion, when and how and what size of deal would it have? When it comes to how, that's already initiated, and from 2025, and package deal size will be larger than existing ones. So this is our goal. Of course, we are not working with anybody out of big pharmaceuticals. Some pharmaceuticals like to have the anti-cancer agents based on ADC, regardless of having their own proprietary ADC or not. So this is our aim in terms of big package with big pharmaceuticals companies. Here is the conclusion of my presentation. If you have a good understanding, you will understand what would be our direction of technology transfer, logically thinking. So clinical stage ADCs, they are already showing differentiated advantages like GOT and RRO and through other ADCs. So the clinical validations suggest that the other ADCs would also be successful, and that is happening in reality. And then the BioBest will be one part of the ADCs. A new payload and AICs will also be part of new ADCs. So the bispecific or ADIC or additional values through innovation, and when everything is combined, as a result, the overall licensing value will increase significantly, and BioBest and the big package deal has higher possibility. In other words, instead of single assets, actually, of course, we'll continue to work on single assets and platform deals we have done so far. And on top of this, we are going to take bio-based approach deal and big package deal. They are going to be proceeded together in a combined way so that we will continue to have the phase 2 or phase 3 clinical trials pipelines, and that will be going on as planned. Due to time constraint, you did not show us the later slides, but I think this is important, so you can show us. Actually, I'm done with my presentation. I'm working in Boston, as some of you already know. So I'm the CEO of a subsidiary in Boston. And I have never introduced ACB officially so far, so I'd like to spend 2 more minutes. So when it comes to ACB, representing antibody chem, which is antibody and chemistry, is part of the company name that is based in Boston. And me and BD personnel are working together. So this is a hub for BD. And LigaChem's IND is being prepared here. So you know that human resources really matter, and ADC company is not an exception. So we have a total of 120 researchers. Many people are curious how these people can manage that many pipelines over the past 10 years. Of course, we have secured new human resources and enhanced internal capabilities. But given the ADC experience, if you want to hire people with more than 20 years of ADC, that is very rare. That's not existing in Korea because ADC history is very short. So we are recruiting talent from the US. For example, Bob Lutz is a chief development advisor who is working with us, Rodrigo, and chief America office. So they're working on the the clinical trials. And Maurice is responsible for CMC until approval, and then the Rexon is responsible for the pharmacology. Yeah, their reputation, experience matter, but I like to say that many other companies are receiving advice from great experts. But usually, they are flying once a year, or they are having in-depth conference about half a day. But the mode of working here at our company is that they are considered as one department at our company, so that we have several meetings a week and we exchange lots of emails with each other a week. So they are looking like our team members. So these are the leadership, and also here are the working level directors. So most of them have the work experiences with 10 years. And Steve and Joe and Matt, all of them are experienced experts specialized in ADC. Apart from these 3 people, we hired 2 more people. One is from the Immunology, the other one is from Big Pharma. Their joining us was confirmed. So all of these people were already working with either Big Pharma or ADC company with ADC with other companies. So what I'm trying to say is that they are elites. But why do you think they joined LigaChem? They have a belief in the future value of LigaChem as experts. So that the BD process starts with R&D, and these are the R&D people, and they are ADC experts from Big Pharma or the Immunology company. Thank you very much. So we are running out of time.
Let's take a dive into his presentation right off the bat. I'm sure it must be really tiring. So all those speakers who have talked about this topic so far majored in STEM, but my major is humanities and liberal arts. So this is the snapshot summary of our discussion so far. So our IR team will deliver this summary to you in a more digestive manner, so I'm going to skip this. So let me give you an overall overview of this vision. Personally, with Mr. Kim Young-ju, we started out together from LG, and we've been together about 39. And at LigaChem, we've been together for 29 years. So those STEM major experts talked about all this. In order to do this, what do you need? Funds and human resources. So what I do is to provide funds and human resources. So over 10 times, I have funded over 900 billion won. At our company, we currently have 560 billion won in cash, and we are moving in a very smart and quick manner. So smart and quick are not two different words. You can actually do work in smart, but at the same time, and quick fashion. So in order to move the preclinical stage to clinical stage, so you need a lot of money. You need about 60 to 70 billion won for the preclinical period and in transition. So we do have a lot of cash. So we can afford to conduct all these clinical trials. For R&D, how much do we invest? Last year alone, we invested 150 billion won, and next year it will increase further. And as we transition from preclinical to clinicals, it's going to require more funds. So we received investments from Orion, but it's not going to dry out because we are going to make more money. So we will make more money than our investments. Of course, we will become a company who turns big profits. So don't worry about the funds or financial situations. Currently, our market capitalization is about 4 trillion won. So our goal is to go over the Orion's market capitalization, which stands at 4.3 trillion won. That way we can actually return to their investment. We can return their favor by delivering more ROI to them. For the past year and a half, Orion, you might think Orion might have been intervening in our business as a big investor, but I can say this with confidence. For the past 1 year and 3 months and 4 months, we are enjoying great freedom because Orion leadership is really gentle and they ensure freedom for us. as long as we do our job right, they are not going to make an intervention. Because we are doing so well, Orion has deep trust in us. So that is the source of our immense freedom. So we are going to become a global company. Please encourage us and support us. Thank you very much. So you cut your 10-minute speech to 3-minute speech. Thank you very much. We were supposed to end our event at 3:50, which is now, but we didn't take many questions. So we are going to extend our event by 10 minutes to 4 p.m. So our event will last until 4 p.m.
So we've got a number of questions, and here is the first question pre-registered. So I have already explained this as part of my presentation. That's already been addressed. And here is the next question.
I think I can address this question today. Regarding this question, I think I've already gave answers twice. This morning, I even said China-based ADC tsunami. So without technological innovation, you can get some progress for ADC. So if you develop multiple ADCs, some of them can be really good. So as a buyers, you might want really great ADCs with great clinical outcomes. So the absolute number of ADC trials has increased. So as I said earlier in the morning, the number of clinical trials is definitely high in China, but we cannot catch up with China in terms of number, but definitely we will increase the number of ADC clinical trials on our side. And within 3 years, 4 years, we are going to increase the number of candidate drugs to 20. If half of them become part of the clinical trials, if half of them enter clinical trial phase, it will be a big success. So we are making differentiation from China in terms of technological innovation. In China, there's no technological innovation. They just gives a little technological tweak or modification, or they just combine or assort existing technologies together. So regarding platform technology, I would say this. For the past 5 to 6 years, we have transferred technology at least one time. In one case, you can imagine my desk. Right now on my desk, there's a big stacks of contracts which are negotiating fiercely, and also there's a due diligence paper for certain company, and we have signed a CD with a certain company. If we don't work like this, we can't actually achieve at least one technology transfer. So once we complete one contract, there's another contract about homepage or platform technology transfer. So consecutively, continuously, this type of work continues. This is it. Next question.
Yes, about a couple of the licenses. I already answered this in the previous question. One of the features at LCB, we are never convinced after going through one deal. Let's say, for example, it can be reversed on the last day, so we are never confident, but we have continuously the maintained deals. So our goal is same, but we do not guarantee. And platform deals and drug deals, their strategies are the conflicting. but I'm not really sure. I do not think I understand the intent correctly. For example, we are developing a drug for target A, and there is a company who want to have a platform technology transfer. They also want to target A. That is not easy, but we are trying to find a solution. For example, we like to use payload A, and then would it be okay to give you B only? For example, there is another company who signed deal for the platform, but if they are already working on the same target, it won't be easy. But the possibility of conflict is very low, but even in the case of conflict, we have a solution. And as part of the platform, I already talked about this in my presentation. We have 2 remaining questions. Please be brief.
Third question. It's very hard to briefly answer this question, so I get lengthy when addressing your question. It's hard to give you specific details, but let me say this. Number one, the clinically validated target and novel target. So we are going to maintain our pipelines for those 2 types. In terms of clinical settings, there's a disease, there's a cancer with a large market, with huge medical unmet needs. It seems like we have a lot of anti-cancer therapies, but the largest markets are still lung cancer and breast cancer markets. For colorectal cancer or pancreatic cancer, have we given up on that? It's very difficult, but if you make a success, it will be a jackpot. So we continue to conduct our research and clinical trials on them. So broadly, we are trying to strike a balance. So since we have a lot of pipelines, we don't just focus on 1 or 2, just we focus on broad spectrums of cancer. Last question.
In order to gain competitive advantage, so we are already competitive. We took the first place, so I'm not really sure what is the intent of this question. So you can give additional explanation to the person who asked the question in private. With this, we'd like to conclude today's event. I'd like to ask everyone online and offline for joining us today in order to become the global top ADC at the company. It has been a very meaningful event to share with you our version and differentiated technologies and sustainable growth. With this, we'd like to conclude today's event. Thank you very much once again. Thank you.